actually intensive effort and partnership and support from everyone is we deeply appreciate. Thank you so much.
And thank you for the opportunity, Taslim.
No, thank you, Asif. It's a pleasure.
Good morning, Jayashree. This is Nandita from CCAMP.
Good morning, Mr. Varun.
Good morning. How are you?
Oh, we are good, thank you. You can hear us, right?
Yes, very clearly.
Okay.
Morning, Jayashree. Morning.
Hi, Debolina. Am I audible?
Yes, Jayashree, you are. Yeah, and we have Nandita
I
also from
can't hear
CCAMP.
you. Let me disconnect and join again.
Oh, you okay? Good morning, Mr. Karthik. Can you hear me?
Yes, I can. Good morning.
Okay,
Okay,
great
thank
to
you.
see you here. Yeah, thank you.
Yeah, morning, Karthik. Nandita from CCAMP.
Okay, ठीक. Perfect. Thank you.
Yeah. Debolina.
Yes, Chandrika.
Shall I start recording?
Just a minute, we'll start. I think there are 2 more juries to join.
Yeah, we're
After
still
which,
waiting for Sunila
Miss-
Thatte.
yeah, and Miss
Ashwin.
Padmaja. Yeah.
Ashwin
And-
Kashikar is here.
Yes, he is here. He was
Oh.
here. Did he, Dr. Ashwin? Did he drop out? No, he's here.
He's here.
Dr. Ashwin is here.
Yeah, morning, Dr. Ashwin.
We'll wait for Miss Padmaja, Mr. Srimuthu, and Miss Sunila Thatte.
Sunila Thatte, yeah.
Miss Padmaja is here. Good morning, Miss Padmaja.
Good morning, good morning.
Morning, Nandita from CCAMP.
Hi, how are you?
Good.
Hi, Padmaja. Hi. Morning.
Hi, everybody. Morning, Nitin.
Morning.
Chandrika, do you want to check with Miss Sunila?
Yes.
Thank you.
Hi, good morning. This is Sunila. Am I
Oh, you
audible?
have joined. Yeah,
Okay,
yes.
good
Yes.
morning.
Good morning.
Good morning.
Very good morning.
Nice to see you.
Yeah, very nice. I've got some issue with my video. I'll try and fix it.
Sure.
But
No
I
problem.
can see the deck and I can also hear you all. Good morning,
Sure.
everyone.
Good morning. So I think most of us are here.
Yeah,
what about
we can
Mr.
start.
Donna and Srimuthu? Are we
Uh,
expecting them?
yeah, they're supposed to join. Nandita, you can.
I'm actually on.
Oh,
Okay, Donna,
okay.
you're there?
Donna, you're there? Hello. Yeah, lovely
Good
to see
morning.
you after a long time. Good morning.
Donna, Srimuthu would be joining?
Yes. Mm-hmm.
Okay.
I'll send him a message.
Sure, that's fine. Yeah.
Yeah. So we have all the participants also.
Super.
Chandrika? Okay.
Yes.
So should we just start? And
Yes, Nandita, we can start.
Start because we have a tight schedule.
Right, yes.
So good morning, everyone. And it's really wonderful to be here and see all of you. And welcome to India's most awaited event in the Bio Startup Calendar, the grand finale of the National Bio Entrepreneurship Competition or NBEC as we know it. CCAM, the Center for Cellular and Molecular Platforms, is a Department of Biotechnology, Government of India initiative, and is today one of India's most exciting life sciences, innovation and entrepreneurship hub. And over the years, we have created a vibrant ecosystem to foster deep science entrepreneurship. So as most of you know, we fund startups. We have a state-of-the-art incubator. We mentor startups. We conduct accelerator and advancement programs, run entrepreneurship development programs, provide access to high-end technology platforms. We have Discovery Innovation Accelerator that takes academic discoveries to industry readiness. We also have an IP and tech transfer office, as well as a Center of Excellence for Agri-Innovation. The BREC Regional Entrepreneurship Center, BREC, was set up at CCAM with the objective of amplifying bio-entrepreneurship across our country and working closely with entrepreneurs to transform their ideas into sustainable commercial ventures. Our thanks to BREC for their enormous backing and providing us with this wonderful opportunity. So under BREC, we conduct the National Bio-Entrepreneurship Competition every year. NBEC, as most of you know, is a nationwide competition to identify, attract, and nurture bio-entrepreneurs working on novel and scalable business ideas with significant societal impact. As you know, this year we have two tracks for NBEC, one a student track and one is a startup track. All of us here are in the startup track. As we speak, the student track is happening in parallel. So we are delighted to have you all join us in the 4th edition of NBEC to celebrate the bio-entrepreneurial talent that this competition has uncovered from across our country. NBEC, as you know, is a collaborative effort with unflinching support from our partners from the biotech industry, investors, entrepreneurial ecosystems, and industry associations. It is with a sense of pride and gratefulness that we present our partners for NBEC 2020. Our prize partners are Win Foundation, Biocon, Ankur Seeds, L'Oreal, Origene Discovery Technologies, Persistent, Pfizer, Novozymes, IQVIA, Anthem Biosciences, BREC, K&S Partners, BioNeeds, and IdeaSpring. Our investment partners are Social Alpha, Molecular Connections, Sangam Venture, Indian Angel Network, Enzia, and 91 Springboard. Our mentorship partners are Biocon, Kotak, and Molecular Connections. CNBC TV 18 is our exclusive media partner. Our associate partners are CIIE.co, RT Lab, and ISBA. Your Story is our online media partner. CII, Confederation of Indian Industry, is our Apex Industry Association partner. Our cloud partner is AWS Activate. Health Venture is our digital bootcamp partner. And our outreach partner is Hello Tomorrow. Thank you very much for your partnership. So we will now begin with the pitches in a short while. But before we begin, let me just tell you how we have reached here today. We have received over 3,100 applications from across the length and breadth of our country. After internal and external online reviews, which were conducted by domain experts, 300 applications were selected for making presentations to the jury panel. We had 26 jury panels for the presentation round, and each jury panel had 2 panel members. From the presentation round, about 54 startups were shortlisted as pre-finalists, and they participated in a boot camp where they were trained on building a value proposition, developing a business model canvas, and pitching. These 54 startups then presented to a pre-final jury, which selected today's finalists. So our grand jury today will select the winners of the NBEC 2020. Let me introduce you to our grand jury for today. We have Mr. Nitin Deshmukh, founding member and senior advisor, Kotak Private Equity, Kotak Investment Advisors. He will be the chair of our grand jury. Dr. Murali Ramachandra, CEO of Origin Technologies. Dr. Osman Shahi, Open Innovation Manager, L'Oreal. Dr. Ashwin Kashikar, General Manager R&D, Ankur Seeds. Dr. Sai Seturaman, Head R&D, Pfizer. Ms. Sunila Thatte, VP and Head R&D, Solutions India, IQVIA. Dr. Ashish Paradkar, Director and Head R&D, Novozymes. Mr. Paresh Vora, Director, India Operations, Win Foundation. Mr. Karthik Chandrasekhar, Director, Sangam Venture. Ms. Jayashree Kanthar, Co-founder and partner at Enzia. Ms. Padmaja Ruparal, Co-founder and President, Indian Angel Network. Mr. Vaikunt, Ms. Jayashree, Ms. Padmaja. Mr. Varun Chawla, Co-founder, 91 Springboard. Mr. Vaikunt, Head Corporate Development, Molecular Connections. Ms. Donna Lekhi, CFO and Co-founder, Health Venture. Mr. Sri Muthu, CEO and co-founder and managing founder, Health Venture. Mr. Vipul Patel, partner, Seed Investment, CIIE.co. Dr. Taslim Arif Sayed, CEO and Director, CCAMP. Also part of the jury were Mr. Ajay Bharadwaj of Anthem and Mr. Manoj Kumar of Social Alpha, but due to some personal reasons, they were unable to join us today. So all the jury members have been sent a 10-minute video recording of each participant's pitch in advance. So today the format of pitching will be each finalist will make a 2-minute elevator pitch and then will be asked questions by the jury. We have a large jury panel, so we will take 2 or 3 questions from the jury. And as we move on to the pitches now, I would request just maybe a couple of minutes so that we can move over to the pitching session. And if we are all set, our first presenter will be Mr. Manohar Sambandhan. Debulina, may I request you to?
Sure, Nandita. I'll start sharing my screen with the PPTs and Manohar can.
Yeah, Mr. Manohar, you have 2 logins. Can you please rename one of the logins?
Sorry, I'm.
Okay, fine.
That's fine. That's fine.
I got it.
So I hope everyone can see my screen.
Yeah. Can I start?
Just a minute.
One moment, Manohar. We have 2 minutes to. You have 2 minutes to pitch, as you know, and jury can ask questions. What we would request the jury, as I said, since we have a large jury, maybe you can just state your, after his pitch, you can just state your name and ask the question. We will just take 2 or 3 questions, and maybe for each, we can take turns in asking questions to different panelists. You've been given your scoring sheets, so we would request that you please do score them as per the parameters in the sheet, so that it just makes it easier for us to tabulate and the scores at the end. So with that, Debulina, would you please?
Dr. Manohar, you can start your pitch. You have 2 minutes.
Yeah, thank you. Good morning, everybody.
We are very glad that I'm part of this BREC grand finale, and thanks for this opportunity to present here. The solutions cotton robo mission, picking mission we are doing is essentially addressing only the peak time labor demand of the farmers during the harvesting season of cotton. This mission does what is called as multi-bloom cotton, which is essentially we have to pick cotton multiple times. We typically, as a farmer, we can pick farmers pick 3 times without damaging the crop, and that's exactly what our mission does. No other mission in the world as of today can do this. Other than this, it also has the quality of the cotton is good, and it also reduces the drudgery of the people who are picking. Essentially, we are increasing the productivity of a human labor here, and that's exactly what it is. It's a solution which uses technologies like robotics, 3D vision, and a farm electric vehicle. You would have seen all that in the image. So the technology we have done can be also adopted or repurposed for other crops for harvesting, and also tasks like precision spraying and even mechanical weeding, which we will do later. These are things which can address the other peak labor demands in the farm labor.
You have 30 more seconds.
Our approach has been primarily to use evolving ecosystems like drones, EV, and robotics. Drone primarily for...
I would request everyone to mute yourself, please, when the pitching is going on. Yes, go ahead. Manohar, you can go ahead.
You can unmute yourself.
We are using the ecosystems of drones, EV, and robotics primarily for multiple reasons. The drone is the one which is driving the motors cost. The robotics is where we are using our software and the camera technology, and the electric vehicles where we...
Time up.
Okay, so we are open to questions. Judy, I would request you to unmute yourself and state your name and ask the question.
This is Sai with Pfizer. Just a quick question. Is it, I saw in the video that you have an operating model. Is it already deployed in the field?
Yeah, it is on the testing. We are running a two-arm robo mission now in the field. Still not commercial. Our funding requirement on funding is essentially to commercialize it. It's not commercialized, but we are testing it on the field. We would be testing it in one of the large seed producers of cotton also in the coming months.
Okay, and how does the productivity compare to human labor?
One robotic arm can pick 50 kgs per day, and one mission can. We need one operator to run this mission. The commercial mission we are going to build is with four arms, so it will pick 200 kgs. It is 4x to 5x of a single human productivity.
This is Padmaja from IN. How do you plan to sell these machines?
See, the cost model of this
strategy.
The cost model is such that a 5-hectare farmer should be able to afford this and recover the cost in 3 years. But in the longer term, we plan to sell it to farming as service companies like EM3 and FPOs. So current cost model, the farmer will recover the cost if he has a 5-hectare farm, and he will recover the cost at 3 years. At yield is 1,200 kgs per acre, and cost of picking is 10 rupees per kg.
Jury has 1 more minute.
Yeah, I think the ability, you should think about the fact that the ability of the farmer to spend so much money on capital equipment is going to be difficult, and even EM3 kind of companies are leasing models rather than buying equipment. So I'll just leave it at that.
Yeah, thank you.
From a scale and marketing perspective, do you think that this is a solution that is global in opportunity?
Definitely, yes. There is, other than US, nowhere else this missions, cotton is can be used in West Africa is one nation which I have been tracking. So there is a whole opportunity in West Africa and definitely even in China, but I'm not really close to Chinese market. But the solution, if we readopt it for other horticulture produce, there is a lot of opportunity. I am actually, some of the companies are pursuing me for other.
Yeah, time up.
Thank you, Manohar. Thank you for the presentation. Thank you. I'll invite the next person, Nikhil Das. Nikhil, you can.
Yeah, good morning, all. Hope my video is clear.
Yes, yes. Okay. So, Nikhil, your time starts now. You have 2 minutes to pitch.
Yeah. So myself, Nikhil, I was the ex-CTO for Havels, and I had built a company which is into LED lighting. The first LED lighting company in India, from 0 to 450 crore I have built. So now this is my second venture. So along with me, this particular venture is being supported by Anshad Aminsa, who is a AI and ML expert. And we have a chief consultant, Virab Sir. And a similar way, like we have a group of AI and ML engineers and embedded team. So basically, like here, like what we do is understanding like the photometry and like making it usable for analyzing a seed. So here, like we can analyze various varieties of seeds and can like tell what morphological parameters it has got and how it can be distinguished between like various genotypes. So here, and one key highlight here is like the aim is to like eliminate this GOT test. Grow test is something like which requires a lot of land and water. Like that particular thing is being like can be eliminated with this kind of a visual evaluation. And another key point is like making this particular industry transparent. See now,
30 more seconds. You have 30 more seconds.
Yeah. Now farmers doesn't have any kind of idea like whether the seed what he is giving is of perfect quality, like because it's all based on the seed companies what they are giving the feedback. So here, like he'll be able to detect it using a mobile application, which we are providing free of cost to the farmers. So ultimately our customer is going to be the seed industry itself, like wherein like this particular hardware equipment will be sold to them.
Yes, time up.
I'll open to questions from jury, please.
This is Murali Ramachandra from Origin. So my question is, what are your competition in India as well as outside of India?
See, we have basically globally two competitors. Like CDX is one company and Videometer is another company. But both these systems we have evaluated, and we could like ours is giving better results. Right now we have about 19 varieties of paddy and 8 varieties of cotton and 7 varieties of watermelon. These things have been identified. So whereas like cotton identification is like cotton seed identification is not possible in seed vision also.
So, Mr. Nikhil, can we segregate between the female parent and the hybrid seed?
Yes, that is one highlight. Like, yeah, we have that kind of advantage. We have already deployed it, and the POC is already detecting these parental seeds.
And when you said that it can be used by farmers, so where you see the value for the farmer in your?
So basically, today he doesn't know like most of the things like how it is giving results. So basically, this particular mobile app which we are providing, this system is being taught, and the AI engine which is analyzing the seeds in a seed industry, that particular data is being used for the helping the farmers to evaluate. So that way he can identify not only the broken seed and those things, like he can even start identifying like the quality of the seed, like the breed varieties. So he'll be getting paid for the right value.
And can we, just last question from my side, can you help us in sorting the seeds? Like if there is a lot which is let's say 90% pure but 10% impurity, so with your system can we remove the bad ones from the good one?
Yes, yes. That actually, with very small integration with a regular seed sorter, our system can provide input and do the sorting purpose. That is one of the key highlight of the solution.
We have last minute.
This is Sunila from IKEA. So my question is, in the short term and medium term, what are your scale-up plans?
Initially, like we are targeting about 5 seed manufacturing companies in India. Like once it is proven over there, like for their seeds, like we are planning to go for a global expansion. Wherein like we are targeting few VC funds also. I'm also like part of a tech evaluating partner for few VC organizations globally.
Thank you.
Nikhil, from the visual, have you established equivalence with the current way of evaluating the seeds, which is you say that they have to actually grow it and then see?
Yes, we have evaluated, and there is a study which is going on from past 6 months, which shows like considerable improvement in our AI algorithm. Like the data what we have captured is like giving almost like 93% efficiency. I'm the 93% accuracy.
Yeah, time up.
I hope the jury has no more questions.
Jayashree, did you have a question?
Yeah,
quick question. No.
Just wanted to understand, is this spectral imaging used by other big seed companies today, Nikhil?
It's not only spectral imaging. Like as I told, my background is into photometry, so I understand light. So that's where the things come in. So,
thanks.
Yeah. Okay.
Thank you, jury, and thank you, Nikhil, for your pitch. We'll move on to the next person, Kalyani Shinde. From
Hello everyone.
Innovations. Hi, Kalyani.
Hi.
You'll have 2 minutes, so you can start now.
Yeah. So hello everyone, this is Kalyani. Me along with my team, we are working towards redefining smart warehousing in agri-tech sector. I personally come from Lasalgaon, it is known for Asia's largest onion market. And the vision was to help farmers because we have seen in the entire onion ecosystem, the fluctuations and the gap between demand and supply and how that is affecting the ecosystem. So we sort of have seen the post-harvest wastage which is happening. So we are working into the warehouse sector specifically, and we are one of the companies who's actually specifically working on a problem which is what the farmers are facing. We have created an IoT-based hardware device which helps create the warehouses into smart, which helps converting the warehouses into smart warehouses. It does real-time microclimate monitoring and it does early identification of wastage. This is actually the USP of our product, which is a farmer's need. So we are trying to help farmers with identification of where exactly this wastage is happening. Our target audience is the farmers with individual warehouses. We are based out of Nashik, where 80% of the farmers cultivate onion as a crop, and probably are planning to scale to Madhya Pradesh, Karnataka, and other states which are coming up with onion production. Other than that, we are also looking at FPCs and government organizations, where we are looking at collecting a lot of data on field, which will probably be used, and we can monetize that data in the future. And we're planning to create a value chain around this.
30 more seconds.
We're also planning to scale to other crops. We've been getting requests. So we are starting to explore poultry industry where the similar kind of a product can be used. And we're looking at more perishable items and which probably are related to warehousing sort of. So yeah, that is probably about it. Thank you. Thank you so much.
Thank you, Kalyani. I'll open to questions from jury now.
Hi, Kalyani. This is Vaikunt from Molecular Connections.
Yeah.
Just wanted to check out what kind of independent validations are done on this device and the way it is used in the warehouses so far?
Yeah, so one is we have done this validation with the users themselves on field. We've done with multiple farmers in their own warehouses. Other than that, we have had our domain advisors or domain validations from people like Sayadri Farms, which is one of the largest farmer producer company in the country. And then we have our domain advisor on board, which is DOGR. So it is Directorate of Onion and Garlic Research Center. So these are the government research organizations specifically working in this sector. So they have been validating our research as well as product, and we have conducted a pilot in their own campus, in their own warehouses. So sort of this is how we have come so far.
Okay, thank you.
Kalyani Jayashree from India. What is the unit economics, and is my understanding correct that your projection is 1 crore revenue in year 3?
So it is, yeah, obviously. So this is more like one. So the cost of the product goes somewhere between. It is a range because there is medium, low, high. It depends upon the warehouse capacity and things like that. But what we are looking at is the unit economics actually depends on the value of the crop which is being stored in the warehouses. It's not about the warehouse; it's a value worth. So even if you take a minimum warehouse of like a 40 metric ton, the value of the worth crop is around 8 to 10 lakhs, which is stored every year in that warehouse. And we are planning to save 20 to 25% of that wastage with the devices which is being deployed. And even the highest cost can be around 1.5 lakh rupees, which is also over a period of 5 years, which can be divided. So it's like more, it's in the 2 years, they can have return on their investment, but it is going to give a long-term return for the farmer itself.
So this is Sunila from IQVIA Kalyani, and if I may ask a follow-on question on that. So from the economics of setup, what's your feasibility from the affordability point of view?
For farmers, are you checking for the farmers, ma'am?
Yes. I mean, for the farmers to install this, yes.
Yes.
You have 1 more minute.
Yeah. So currently we are actually, we have had validation from some farmers where now this becomes a very tricky part for us, and we are also validating it more because onion ecosystem is very fluctuating. So we need to figure out that model. So what we're trying to do is we're trying to get micro finance partners or who are going to be with us, working with us to get this deployed for them, because this is a hardware product. So we also are going to need the funds in hand before to scale to various geographies, and they come in later stage as per their crop. So that's how probably we're planning to go with it.
Would it be accurate to say, Kalyani, that the way you're reducing wastage is actually being able to control the environment better, or is it what is you mentioned something in the video about sensing?
Yeah. So are we not controlling the environment? Yes, there are two models. With individual warehouse, we are not controlling environment. There we are helping them just monitor or identify what microclimate conditions are there, because they use some frugal techniques to control temperature.
Time up.
It's okay. You can. Yeah, you complete.
Yeah. So, so with the individual warehouses, we actually do sensing of where the wastage has started. So onion as a crop, it emits some gases out of it. So with the devices which we have deployed, we can sense where the rotting has started or where the wastage has started in the exact warehouse. Currently, the farmers are doing it based on the smell coming from the crop, based on the level going down. So these are very conventional ways to identify wastage. And in the second model, where I also talked about community warehouse, in these warehouses, this is like an end-to-end solution, where we are working with a microclimate control structure in partnership with Tata Sayadri Farms.
Thank you, Kalyani.
Thank you.
Jury, are we good? Then we can go ahead with the next person. I'll invite Sonali Khandra. Hi, Sonali. You have 2 minutes for your pitch, and I hope you can see the slide.
Yes.
Okay, you can start.
Can you hear me?
Yes.
Okay. Thank you. Good morning, everyone. I'm Dr. Sonali Khandra. I'm a PhD in nanotechnology, postdoc in microbiology, and trained in technology licensing. Our vision is to prevent disease by democratizing gut immunity through natural yet innovative solution. Presenting Nano LF Pro, a unique formula based on nano encapsulation technology developed through extensive research during my doctoral work. Before I speak more about the product, I would like to spend 30 seconds on IBD. So IBD is a chronic inflammation in the gut. 10% of global population suffer from IBD. India is second largest after USA. This occurs in early stage and remains throughout the life, thus hugely impacting patient education and employment, and also substantially reduces their quality of life, as it affects physically, mentally, socially, and economically. Now, coming to the current treatment limitations, they all have single mode of action and does not provide complete solution. Hence, always require a drug regimen, either in combination or one after the other. Drug non-compliance is a huge problem in IBD because of the multiple medication, their associated side effects, increased drug resistance, and long-term. Moreover, it is not economically viable to all the sectors of society, as it requires lifetime management. So with this background, let me talk about the proposed solution, Nano LF Pro. It's a synergistic combination of lactoferrin and probiotic delivered via a smart nano-encapsulation technology.
You have 30 more seconds.
Lactoferrin is a natural protein found in cow and human milk, known for its multifunctional properties such as anti-inflammatory, immunomodulation, antioxidant, and antibacterial. Additionally, lactoferrin promotes colonization of probiotics in the gut. It's an FDA-approved and GRAS molecule. Nano-encapsulation delivery guarantees that probiotic and LF protein survive stomach acid, arrive in intestine, and remain available for longer time. So in a nutshell, it's a holistic approach to provide end-to-end management of IBD, and our team has all the expertise to make this feasible. Thank you very much for the opportunity and looking forward for your questions. Thank you.
Thank you, Sonali.
Hi, Sonali. Yeah.
Yeah, this is Paresh Vora. Can I ask a question?
Yes, please.
Yeah. Have you thought of these applications in any public health in terms of mother and child nutrition? And then second question is, what is the current status? Yeah.
Yeah. Absolutely, sir. This has a very broad spectrum. We wanted to start with gut immunity. And as you must be aware that gut immunity is actually related to all the mental health issues. And lactoferrin is already known for its application on neonatal sepsis. So childhood health is absolutely one of the most important application of this formula. It is already lactoferrin is already known to help neonates during the sepsis. So it's definitely one of the biggest applications.
And what is the current status? Oh, sorry to.
Yeah. So the second question is the current status. The technology is developed. It has already been tested for targeted delivering other anti-cancer drugs. I have several publications on that. This is my PhD work. Now we have to establish its animal efficacy and safety. Technology is already developed and been tested.
Why is this better than, say, an enteric-coated tablet that you could deliver?
Sorry, could you please repeat?
I said, why is it better than the enteric-coated, which is a proven technology to produce tablets that can go past the gut?
Correct, sir. Absolutely. Enteric coating is one of the way to pass stomach acids, but nano encapsulation provides many other things, which is targeted delivery. It also reduces the entire toxicity. So in a way, it gives you a much more customized way to deliver the medicine.
Sonali, this is Ashish from Novozymes. On the same line, demonstration of efficacy in very diverse human populations is the most challenging aspect of these technologies. What is your plan to do that?
Definitely, sir. We have so once the technology established in IBD, we wanted to start with IBD, and we would go for a clinical trial. We have a clinician in our team. He's very efficient. He's a gastromedical enterologist, and we are actually following his suggestions on how to go about the clinical study design. So we would be following all the guidelines for the clinical trial in a larger set of which would be actually a statistically significant population.
Hi, Sonali. This is Taslim Arif Sayed here from CCAM. Just want to understand your technology arbitrage, because lactoferrin has been known and even was considered once in COVID time, and probiotics are anyhow fairly known. Yes. Can you just provide one layer detail of what is the technology here? The combination I can understand, but there are available natural substrates if we are talking about. So what is that you're doing add-on that you can patent?
Sure. So we are the synergistic combination is something which we are thinking of getting patent on, because there is no combination available as such. Yes, lactoferrin and probiotics separately available, but again, the increasing the bioavailability is something which is very, very important, and that's the reason lactoferrin is not really been realized its potential. So we are trying to increase the oral bioavailability by putting it in a nano encapsulation. The technology is it's an emulsion method, and the technology is also, the know-how is also something which would be able to patent. Sorry, did I answer your question?
No, thank you. Thank you.
Yeah. Time up.
Thank you, ma'am. Thank you, everybody.
Thank you, Sonali. Thank you, Judy. We'll go ahead with the next person. I'll invite Srishti Batra.
Hello, can you hear me?
Yes, Srishti, we can hear you. You can see the slide, right?
Yeah, I can see the slide.
Okay, Srishti, you can start. You have 2 minutes.
Yeah, a very good morning, everyone. We are QSense Labs, and we are transforming fresh food supply chain with data. Globally, 40% of the fresh produce is lost post-harvest. India alone lost about $15 billion in 2019, and globally these losses are over $1 trillion. Quality and freshness assurance is a big problem in the fresh produce retail, owing to manual decision making relying on intuitions, leading to high inventory losses and suboptimal margins. We are solving this problem. QSense is a deep tech end-to-end AI-driven IoT platform for fresh produce quality management. Our patent pending technology integrates near infrared spectral sensing and olfactory sensing that sense of smell to quickly and accurately assess the quality and freshness of fresh produce, thus providing a holistic solution. We help in reducing wastage by over 30% by increasing the margins from the same produce, increasing customer satisfaction, and bringing in transparency through data. We are currently serving 4 customers, including India's biggest retail chain, Reliance, and a leading e-commerce player. I am Dr. Srishti. I hold a PhD from NCBS TFR. I have over 11 years of research experience across top universities of the world, including University of Minnesota and Lund University. I specialize in olfaction, the key technology that we are developing over here, and I bring in great experience in building ML pipelines and big data analytics for biological data. My co-founder Rubal has over 8 years of experience building IoT products from scratch.
30 more seconds.
She built India's first smart bulb IoT at Cube 26, and prior to QSense, she was working with Hevels in their R&D division in the smart home range. Together, we started QSense in 2019 with the aim of revolutionizing the fresh food supply chain with data. Thank you, and we can take questions.
Hello, Srishti. In terms of olfactory sensors, actually, can you let us know how big is your library? And also, in terms of actually sensor accuracy as well as sensitivity, what is the effect of temperature and humidity?
We are actually integrating temperature and humidity in our spectral sensing as well as in our olfactory sensing in building the ML models that we are building. And we are seeing some dependencies on that. And the accuracies, so we have one of the devices, the NIR device is currently with the customers, and we are having an accuracy of about 95% at the customer's end. So, and in-house, we go up to even higher in control when we are doing inside the office or in the lab. With the olfactory loggers, we are building on models, and currently, I think I would be able to comment on accuracies in coming weeks.
Hi, Srishti, excellent work. This is Taslim Syed here from CCAM. Very quickly, just want to understand in terms of olfactory sensors, would you imagine a completely different spectrum at different places in terms of the chemical moieties that you would be testing? And how do you anticipate that for a regular, standardized sensors?
Yeah, so that's a very important question because each environment is really different. So the approach that we are using is a multi-area approach, which is something that actually our own nervous system employs, which is a combinatorial coding. In that way, we can actually incorporate a lot of multiple scenarios with just a handful of sensors. So we are taking up that approach for it.
Quick question from me, this is Nitin here. Does it also identify gene markers, and what is the pricing that you are following?
No, we are currently not working on the gene markers. We are mostly working on internal features with the NIR spectroscopy, something like total soluble sugar content, protein content. And with the olfactory one, we are mostly looking into the different gaseous components like ethylene maturity, ripeness maturities, those things we are currently testing. We are also looking at a case of pesticide residue, but that is still work in R&D, so I won't comment it now. And the pricing is different for each of the products. So it's a combinatorial product, and you can have a combination of our handheld scanning device with the software platform and the olfactory sensors with the software platform. The costing is different for both. With our complete solution, we actually help them increase their revenue by 10%, the current customers, and we are actually charging just about 20% of it. So they recover the cost within a couple of months. To be like in less than 3 months, they recover the cost. The handled one is currently priced at about 10,000 monthly subscription for the customers that are currently using it. For the handle, it's for these olfactory sensors, it's a little bit on the higher end as the sensor array is a bit expensive.
Okay,
Time up.
Thank you, Srishti. Thank you, jury. We'll go ahead with the next person, Subhash Narayan. I think Phoebe George is representing Subhash. So Phoebe, you have 2 minutes for this slide.
Yeah, okay. Can I start?
Sure.
Yeah. Good morning. The oxygen in the air we breathe is transported through the blood vessels in the form of oxygenated hemoglobin, and we at SASCAN monitor the changes in the oxygenated hemoglobin absorption to screen and detect cervical intraepithelial neoplasia and for biopsy guidance. Cervical cancer is a growing concern in India with a mortality rate in excess of 50%. This is primarily due to the late stage detection associated with the low sensitivity of pap smear, also due to the lack of awareness and reluctance of women to screening. Colposcopy often leads to unwanted biopsies owing to its low specificity, and there is a long wait period for results. In this context, we have developed Cerviscan, which is a novel handheld multispectral imaging camera for fluorescence and diffuse reflectance imaging. This device consists of multiple LEDs to illuminate the tissue and a monochrome camera to capture the images. The captured images are processed and analyzed using a machine learning algorithm to detect cervical cancer from oxyhemoglobin absorption maps. The device has also the ability to locate the most malignant site in the cervix for biopsy so that the underdiagnosis can be avoided. We are planning to conduct clinical studies to validate the device in medical colleges at Manipal and Thiruvananthapuram from next month. Ethical clearances and CTRI registration are in place and approval of CDSCO is obtained. SASCAN is an ISO 13485-2016 certified startup, and we have a 7-member team consisting of scientists, engineers, doctors, and marketing experts with a combined domain experience of 90 years. We plan to market the device.
Your time is up.
Okay.
We'll open to questions now.
I had one question. EVA Colpo seems to be the closest competition. So what is the sensitivity and specificity comparison against that?
Ma'am, we have done the proof of concept in Regional Cancer Center Trivandrum, and we obtained a sensitivity of 87% and specificity around 92% for our device.
And where is EVA Colpo?
We don't have any reported information on that actually.
Oh, okay. Okay.
You have any, Paresh Vora here, any comparison with, there is a Pune-based startup which is working on same thing, and they've also done some trials in some hospitals.
Yeah, that is periwinkle technologies you are looking at, isn't it? Yeah, yes. They have a white light imaging system by which they are similar to a handheld digital colposcope. They have the probe which goes through the speculum into the oral cavity and take the image. But ours is a multispectral imaging technique. Their system is not quantitative, whereas that is again subjective, and they are using some machine learning algorithm to look at the lesion changes with white light illumination. But we use multispectral imaging, and we have a technique of looking at the quantitative changes in the malignant transformation using our imaging technique. That is which we are doing with a monochrome camera and multiple wavelength illumination. So our accuracies are much better, and we can also direct the clinician to the most malignant site in a lesion for biopsy with our technique, which the other technique doesn't have the possibility to do.
Hi, this is Taslim Sayed here. What's up in next 12 months? What are you going to do in next 12 months? How is the product progressing in next 12 months? Is it the market entry? But sorry.
Yeah.
Sorry, I think Phoebe, it will be good if you can take the questions, please.
Yeah, yes, ma'am.
Since you are presenting,
So
thank you.
Yeah, we'll be starting the clinical trials from next month, Jan, and up to June, for 6 months, we'll be doing the clinical validation. After that, we'll be doing the DFM, and by the end of the second year, we'll be trying to reach the market. That's our plan for the next 2 years.
Okay, thank you.
Thank you, Phoebe. I think we don't have any more questions right now. Thank you, Phoebe, and thank you, Dr. Subhash.
Thank you.
We'll go ahead with the next person. I would like to invite Dr. Vishal Rai.
Hi, am I audible?
Yes, Dr. Vishal. So you'll have 2 minutes and your time starts now.
Thank you for the opportunity. My name is Vishal Rai and I am representing Playable Tech today. So I would like to talk about our ambitions for directed cancer chemotherapeutics and how we can meet the global technological demands in this sector. The antibody drug conjugates or ADCs provide a state-of-the-art technology for directed cancer chemotherapeutics. A few years back, the pharma sector noticed an enormous surge in the efforts in this direction, but unfortunately, a significant percentage of initiatives failed at the pre-IND and IND stages. So the big roadblock emerged from ADC's structural complexity that makes the synthesis and characterization an absolute nightmare. So in the absence of technologies for precision engineering of antibodies, the pharma industry was mostly dealing with heterogeneous ADCs, which led to insufficient manufacturing and controls data. So it is well accepted that the synthesis of ADCs with robust control over precision regulates all the factors that are critical for the required characterization data. Moreover, it is essential to resolve the challenges of pharmacokinetics and batch-to-batch reproducibility, along with clinical efficacy and safety. For the first time, our LDM platform and mass spectra technology provides a practical solution to this well-recognized technological demand. Our main focus is to start with trastuzumab emtansine conjugate and come up with a direct competitor for Kadcyla. We are confident that our ADCs have the potential not just to outcompete their rivals, but they provide an excellent opportunity for the access to next-gen biologics.
You have 30 more seconds.
Sure. At present, we have already developed a set of highly homogeneous ADCs with analytical data and in vitro studies, that is setting up new benchmarks. Our team has worked out a clear roadmap, and once we reach the pre-IND or IND stage, we plan to out-license our ADCs. Thank you very much for your kind attention. I'll be glad to take your questions.
Yeah, hi, this is Nitin here. Quick question. In terms of the progress, have you been able to identify some novel payloads and novel linkers for this ADCs?
So in terms of novel ADCs, we are, so the technological platform that we have developed, we have tested it with a variety of proteins, enzymes, and antibodies to test its robustness. But at the same time, we realized that it would be ideal to first take forward an established antibody and a payload so that we can do a direct head-to-head comparison and make sure that at least we have one candidate that has a shorter lifetime to the market. But
haven't reached the stage yet in terms of
right. So, but you know, on the other hand, we have been testing the platform with, we have tested it so far with 6 different antibodies, but at the same time, we are not taking them forward because they will definitely have a much longer time span for the for translating them to market.
And what do you think the IP will be? What do you anticipate the IP will be in this case?
So the LDM platform, which means linchpin directed modification platform, is actually the first chemical technology that provides precise engineering of proteins along with the modularity. And even till date, there is no other technology that can deliver that precision. So that the basic technology itself is patent protected. Besides, we have mass spectra technology that ensures that for, you know, we can now do very unambiguous determination of not just drug to antibody ratio, but exact site of conjugation of the toxins to the antibody. So this is the initial part. And then the linker that we connect, the flexibility that we get, the site of conjugations that we can regulate. This is all something that creates a completely unique space in terms of.
Yeah, thank you.
This is Sunila from IQ. One quick question. So from the, you know, clinical, how much time do you anticipate this is going to take after in vivo studies and all for actual clinical application?
So since we are taking the pre-established antibody and pre-established toxin, the linker would definitely have novel components. So they still will have to be tested. But I would say from a completely novel entity to this, it will be 1/3 of the time. So that's what our anticipation is. So right now we are already in discussion from last 8 months with a partner who would join hands in terms of pharmacokinetics study and animal investigations for the next generation.
This is Sai with Pfizer. So are you suggesting that you're currently in preclinical studies and then you would be followed by?
Sorry, we are, I guess, at least a year away from that. So we have a well-determined library, completely characterized. So the first 2 stages are done with all the required 10 characterization steps. We are done with in vitro assays. And now the time for pharmacokinetics and in vivo assays is there. So that is, unfortunately, COVID has delayed that, but yeah, that's where we are right now.
Yeah, time up.
Thank you very much. Thank you, everyone.
Vishal.
Thank you, Dr. Vishal. I would like to request the next person, Vaibhav Hegre. Can you see my screen? Can you see your slide, Vaibhav? Cambrian Bioworks.
Yeah, all right. Am I audible?
Yes, yes. Your time starts now.
Hi, I am Vaibhav from Cambrian Bioworks. India has 1 test center today for COVID-19 for every 6 lakh people. Today, in rural India, test samples are sent to centralized labs, which are typically located in densely populated areas, often several hours away. This process is slow, inefficient, and just not enough to take crucial medical decisions. We believe that testing must be democratized. This will require decentralized testing by prioritizing near-care testing sites. Presenting the Curio. The Curio is a lab-on-cartridge system that integrates all test protocol steps on a single device. Our solution will enable rural medical facilities who want to provide high-precision testing without requiring extensive hands-on lab staff. By eliminating the need for sample prep and using shelf-stable reagents, the Curio will enable a large network of near-care testing sites. We are using our expertise in plastics, assay and diagnostic product development to build a modular cartridge design. This open design cartridge can be configured to run molecular assays for infectious diseases like COVID-19 and immunoassays to detect conditions such as HIV. This ability to modularize will help us expand cartridge functionality to also check for sensitivities to medications. Our customers will see value in being able to test on disease-specific cartridges that would otherwise require different kinds of machines and protocols. There is no silver bullet to eliminating unequal access to medical care, but enabling people to resolve their own problems is always an effective path. The Curio is an important step in this direction. Thank you for your time.
So Vaibhav, is this a concept or is it a working prototype you've already produced?
So we've created a bench scale prototype already, sir, where we are actually testing individual components of the process itself. For example, with molecular assays, we will be doing an extraction of the nucleic acids. We are fine-tuning these first at a bench scale system. Additionally, the device itself is already able to control operations such as heating as is required by an amplification protocol and detection of fluorescence changes.
Yeah, hi, this is Nitin here. I noticed that you actually mentioned in a presentation that this device, you can do molecular assays, immunoassays, as well as cell-based assays in your presentation that you sent. And you mentioned the price is 4 lakhs. Do you think this is achievable in that, at that price, all the 3?
So a large part of the modularity will come in the design of the cartridge itself. If you are able to sequence the steps of operations, for example, with regards to immunoassays versus molecular assays, we think that detection of color changes and heating steps are easy to incorporate in a device itself. We have significant experience with building machines itself. So we think that the price points that we see currently in the market just don't make sense for what a BOM is for a system like this. So yes, in essence, we think the price is definitely possible.
This is Murli from Origin. To establish the POC, which are the pathogen that you are going after for the first set of demonstration?
We've begun work on COVID-19, sir, and we're targeting the nucleic acid protein as well as the ORF sequence. So those are the 2 targets we're looking at. We're also building it on a LAMP protocol for an isothermal amplification.
Do you have any anticipated cost of this cartridge?
So we believe that we should be able to bring it to market at around 1,000 rupees or under. That is our target price. We feel confident about what it would take to actually make the cartridge. So we believe sufficient margin in what our planned proposal is.
Molbio has been working on this for many, many years and recently got funded. So how are you unique as compared to them?
So firstly, we will make modularity a pretty significant component of how we will build the system itself. So an ability to switch between molecular assays and immunoassays is a pretty strong selling point, we believe. Secondly, Molbio's system actually involves two different devices, involves several intermediate steps. In our case, we will integrate all these operations onto a single cartridge, reducing the actual threshold of skill that a lab needs even further. Moreover, Molbio's system actually comes to market at around 11 lakhs, which we believe is still a little high to be able to really improve the number of installed machines around. So we see significant differences in how we are approaching it and they are.
Yeah, time up.
Thank you, Vaibhav. Thank you, jury. We'll go ahead with the next person. I would like to request Dr. Raj Bihari Tunga. Dr. Raj Bihari?
Yes, ma'am.
Yes. Can you see the slide?
Yes.
Okay. Okay. So you can start. Your time starts now. Yeah.
Good morning, all. Myself, Dr. Raj Bihari Tunga, founder of Cr Biotech. Today, my co-founder, Dr. Vinita, is not able to join us due to some medical emergency. Therefore, today I will represent the Cr Biotech. Our company aims to make life better of masses by solving public health problem. One such problem is quick diagnosis. There is no such quick diagnostic method to detect dengue, chikungunya, malaria at a single go. So as a result, if you see the doctors, as current practice, doctor test one after one, which leads a long time, as well as late detections and delay start therapy. As a result, patient there will lead a lot of burden of expenditure and also morbidity of the patient. To solve this problem, we developed Assure, one rapid diagnostic combo kit that can detect malaria, chikungunya, dengue on first day of symptoms in less than 10 minutes. Our novel vertical flow immunodot technology with gold nanoparticle and the recombinant antigen gives Assure better sensitivity and specificity. Since it is a combo device, so we can detect co-infections easily. The device will drastically reduce the cost, and as a result, it saves the expenditure of the patient, as well as it saves the time and effort of the doctors and pathologists.
Yeah, you have 30 more seconds. 30 more seconds to go.
Okay. Due to timely treatment, life will be saved. Our device is suitable for rural use also because it uses whole blood. There is no other equipment is required. Currently, we are under validation stage, which is ongoing, and we are ready for this clinical evaluation for next stage. That's all. Thank you.
Hi, Dr. Tunga, this is Taslim Arif Syed. Very quickly, so since you have the validation, is it a clinical validation that you have had on patient samples? Have you sent it to CDSCO for the approval? Is it up for manufacturing license? We do know that this work has been happening for a while. So when you say validation, what kind of validation and when are you getting a manufacturing license?
Okay, thank you for the question. This validation is for not for this clinical evaluation. So that this is the pilot study. Based on that, as per the CDSO, CDSO officers suggest us to do some pilot validations, which we have taken the ethical committee approval and also CTRI registrations. and also take the Form 29, that is the test license. We have taken permission from CDSCO, and then we are doing the small scale validations. This after the validations over, then this result along with the clinical protocol we have to submit to the CDSCO, and then this CDSCO, after seeing our this validation result, as per their requirement, that we have to send some sample to their respective lab where they will test our device.
You're doing that.
Device, and,
yeah.
And then they will provide us this manufacturing license for.
Yeah. So Dr. Tunga, this is Sai with Pfizer. Just a quick question: do you see that this kind of detection technology could be applied to other microbes to have early detection of what kind of infection there might be in the patient?
Yeah, see that again we are working on that. That is called DNA technology, and the DNA array technology. The same dot technology we can use. But okay, obviously that depends on what is the pre-treatment stage, how we can do that. So currently we are not working on that, but that is also our thought process, that if we can, because the pre-treatment stage process, if you can reduce that time, then maybe it can be implemented.
Dr. Tunga, this is Sunila. How much, roughly, what timeline for commercialization are you envisaging?
2021 third quarter, end quarter, we are expecting to launch the product in the market.
Thank you.
Thank you, Dr. Tunga.
Thank you, Dr. Tunga. Thank you, jury. I would like to request the next person, Niranjan. Hope you can see the slide, Niranjan.
I think he has dropped out just.
Okay.
Maybe we can allow.
Okay, then Gitanjali Radhakrishnan, I would request the next person. So I'll just tell the jury to keep a note that the next person is Gitanjali, and we'll come back to Niranjan when he joins back. So Gitanjali, you can see the slide?
Yeah.
Okay, so Gitanjali is Aduvo Diagnostics. I'm just telling the jury to make a note of this slide. Thank you. You can start, Gitanjali.
Hi everyone, I'm Gitanjali, founder of Edivo Diagnostics. We address the global wound infection and assessment market, which covers diabetic foot ulcers, surgical site infections, and burn incidences. What we have developed is a first ever multispectral autofluorescence imaging device, which is combined with machine learning on these multispectral images to detect and also classify gram type of bacteria instantly on wounds when compared to a traditional culture test, which takes 3 to 7 days to come back. Currently, the algorithm is tested on close to 900 patients, covering all these different market scenarios I've spoken about. It has 90% accuracy with that of the culture test and greater than 85% sensitivity and specificity. So as you can see, currently doctors can see a white light image, which is visual inspection based and very error prone. But using our device, they accurately understand which are the regions of infection and how it is being progressed over a period of time. We have a patent granted in India. The same technology can be used in other applications, which is in cosmetology, even in cancer diagnostics. And currently we have commercialized in India. We have an ISO 13485 certified facility. We have 2 distributors, one for private hospital sectors, where we have placed 5 devices so far. We have commercialized last December, and we are also have seen tremendous interest from armed forces and medical services with respect to traumatic wounds. Our long-term vision is to partner with pharma companies, negative wound pressure therapy dressing companies, to take this device as a monitoring cum treatment solution, and also understand the overall antibiotic susceptibility in a particular pattern, and to bring down the rate of amputation and mortality significantly. And we are in the process of CE marking. and aiming to enter into European markets, where we also have tremendous interest from Smith & Nephew, 3M Healthcare, to take this device forward. Thank you so much, and happy to take queries.
Yeah, this is Nitin here. Very quickly, what kind of validation have you done for this, Gitanjali?
Sure. So we have
clinical validation.
Yeah.
I mean, yeah. Sorry.
Sure. So we had taken a site in Chennai, which is for diabetic foot ulcers, in Pune for dermatology-related fungal infections, and in Gujarat, Valsad towards surgical site infections. We take a sample using this device, a deep tissue biopsy, and send it to a culture. The doctors take a regular routine protocol, and then we first compared the gram type of bacteria with our device with that of the culture test. Second, we have also identified that culture itself is not accurate. Many times it has come back as no growth, and our device has 85% sensitivity in detecting the presence or absence of a bacteria, and cultures is only 57% accurate in that sense.
It's both gram positive, gram negative.
Correct. And right now it is also trained for Pseudomonas aeruginosa and also biofilm detection.
Okay, thank you.
This is Murli from Origin. So what's your competition? And if there is competition, how do you compare?
Oh, sorry, can you come again? Okay. Our only competitor is a Canadian-based company which uses a single light source. So they cannot distinguish bacteria or have the machine learning algorithm which can fine-tune as and when you get more and more infection images and bacteria associated with it. So which makes us the first ever imaging device when compared to the other devices, especially in the infection space.
Thank you, Gitanjali. Taslim Arif Sayed here. Excellent
Hi,
program. Very quickly, I just want to ask, in terms of what...
Hi, Sashi from Novozymes.
I'll just quickly finish my question. What would be your false negatives in this case? What do you anticipate? Would there be any skin, the remains or something which could possibly give you that? Have you had that?
Yeah. So we have specifically instructed the users not to use it on bone, because bone has the autofluorescence on its own. So currently we are fine tuning towards bone regions alone. With respect to normal flora that is present, the device looks at the infection marker as well. So normal flora does not get into the algorithm when you're looking at the infection marker as well. And it's always advised not to use betadine solution, and also to generally do a saline wash before imaging.
Hi, this is Ashish.
Ashish had a question.
Yes.
Yeah, please.
So two questions. One is your technology able to discern between species and genus level? That is one question. Second is how deep your technology can go from a tissue sense,
Right. Okay. So the light can penetrate up to a distance of 1.6 mm. With respect to genus and species level, right now we say that we could do a genus level classification, but species level classification would require further integration of more sensitive sensors, and it requires a lot of data collection. It's the way to go, but we are targeting to develop an antibiotic susceptibility device using the same technology to close the loop, where not only you screen the infection, but also you know the antibiotic pattern, what antibiotics to prescribe in under 4 hours using the same technology. I hope
Thank
it
you.
answers the
question.
Mr. Osman, last question. Or I think even Sunila has a question. So if you can just be brief in your responses, Gitanjali.
Sure.
Yeah. How do you account for, non-fluorescent if you have any pathogenic bacteria? Is there any plan to detect?
Oh, sorry, for
pathogenic bacteria which doesn't have fluorescent markers?
Okay.
Non-pathogenic
so we know
bacteria.
that. Yeah, so most of these bacteria have a metabolic marker associated with it. So from our understanding, all bacteria fluoresce from the metabolic markers. We need to take into account what are their infectious marker by doing in-house studies before we can replicate on field.
Sunila, you
Okay.
had a question?
My question is, I saw that you already deployed this in certain hospitals, and are you collecting any real life data, and anything you would like to quickly say about the overall efficacy when compared to other similar devices?
Yeah, sure. So there are, like I said, in India, there are no similar devices as a screening tool. So we have deployed in 5 places where every week the image gets synced onto the centralized cloud, where we do not have access to patient information but the spectral images. In terms of efficacy, the doctors have stopped sending the third day culture for a laboratory test, and they are relying on our device. In case of burns unit, they are actively using it for pediatric burns to effectively ensure that they can prevent the infection rate, which usually happens on the 3rd day. So they accurately now know where the sites are. So these are some of the efficacy, I mean, in terms of use cases that we have seen in the field. Of course, we need to do over a year to better understand how we prevent amputation and other real-time metrics.
Thank you. Thank you, Gitanjali. Thank you, Judy.
Yeah. Sorry, Taslim, I could not take your query, but happy to connect offline. Yeah.
We'll go ahead to Niranjan now. Niranjan, hope you're there. Okay, I can see you.
Yeah, I'm there.
I'll
Thank
just
you
put
very
your
much.
slide back. Sure. Niranjan,
Yeah.
you can see the slide, right?
Yeah, I can. Thank you.
Okay, you can start.
So sincere apologies, I got disconnected. There's been some intermittent internet issues today.
That's
Okay,
fine.
so
We can understand. Yeah, you can start.
yeah, let me start fast. So primarily, I'll narrate 2 instances of 2 patients who were impacted by our technology at 2 different settings. Okay, first one is at a remote district center called Burger in Odisha, and the second one is at a large tertiary care center in Coimbatore. Okay, both patients in their late 50s to early 60s presented to ENT and neuro OPDs with complaints of sudden onset giddiness, nausea, heavy-headedness, bulbar palsy, and unilateral hearing loss. Now, one did not have access to advanced imaging protocols, and the other had an MRI which did not show evidence of any kind of an infarct. Now, both were administered our eye tracking based balance eye test in just 5 to 10 minutes, right in the clinical OPD, absolutely non-invasive. Now, both findings pointed to the presence of central pathology, specifically towards early onset of stroke. Now, both the patients were treated right on time and recovered quickly. So to put it in a nutshell, right, eyes are the windows to the brain. There's enough scientific evidence clearly establishing a correlation between movement of the eyes and the happening in the brain and the inner ear. Now, capturing and analyzing these eye movements provide very early stage diagnostic insights into what is happening in a human brain and the inner ear. At Cyclops, we are leveraging the power of computer vision, image processing, deep learning to build robust stackable eye tracking based tech stack, which has the potential to diagnose close to 24 neuro disorders at a very early stage. And we are right now keenly focusing on dizziness and balance disorders, cognitive disorders, dynamic vision screening, and stroke and traumatic brain injuries. Our products are installed in over 260 hospitals and clinics in India and 6 neighboring countries. More than 100,000 patients have been diagnosed on the platform. And we recently received European CE certification as a Class 2A medical device. This opens up large opportunities outside India. And some of our prominent corporate customers include Manipal Hospitals, 4 units, Narayana Health, Aster, Columbia Asia, command and base hospitals run by the Indian Defense Forces, St. John's Medical College, etc., etc. So this is our executive summary in a nutshell. I'd be happy to answer questions. Thank you.
So Niranjan, this would you say that this is a early detection, but then needs to be followed on with the more conventional diagnostics?
Yeah, it depends on case to case. In most of the problems where the scientific evidence is very clear, it can provide decisive diagnosis. But in cases where the scientific findings are equivocal, it needs to be followed up. But it's an excellent point of care tool.
How do you compare on pricing, Niranjan, with obviously some of the imported devices that are there, like Tobii Pro and C-Light technologies, etc. That's also being used today.
Yeah. So see, Tobii is a generic eye tracking platform, and multiple products can be built on it. And till about recently, Tobii was more focused on research-based utilities. Now, specifically, if I were to compare with some of healthcare use cases, like from interacoustics and so on, we are at least about 3 to 4x more affordable compared to them.
Hi, Nitin, this is Usman from L'Oreal. In terms of business model, I mean, you're only focusing on healthcare, or you plan to venture into other mobile grazing for application development or e-commerce, etc.?
Yeah. So, see, our current focus is healthcare because that's where our knowledge and experience lies. But the technology stack that we have built has applications outside of healthcare as well, especially in aviation, in industrial use cases, in sports training. And we are talking to multiple partners in these areas who are keen to license our technology and build applications in those spaces.
Niranjan Jaishriya from India. What is the larger validation studies that you've done?
Yeah, so far we have completed about 5 validation studies, and all these 5 have been published in peer-reviewed journals. And right now we are in the process of actually completing one of the world's largest multi-center studies for unilateral canal BPPV, where the sample size extends to about 450 patients. And this will be published pretty soon as well. And in addition to that, there are 12 more studies that are going on at multiple locations. So the good thing is, since we have 250 plus customers who have already adapted our product, each one of them are doing their own studies, and that kind of amplifies our overall scientific exposure.
Thank you.
Thank you, Niranjan.
Thank you.
Thank you, jury. We'll go ahead with the next person, Dr. Sugandhi Gopal. I would like to request Dr. Sugandhi, if you can switch on your video, and yeah, I'll just display your slide. You can see this, right?
Yes, I
can. Okay, you can start.
Okay. So we are talking about a 100-year-old machine that we regularly use in the cardiology clinic. This is used every day and almost for every patient who attends in the clinic. And I think all of you would have had an ECG at some stage. So you'll be very familiar with the clumsiness, the tediousness, multiple electrodes being applied, clamps being on the hands and legs, and the technician struggling hard to deploy it correctly. And in fact, in this picture, you can see that it's deployed wrongly, and it could even give a sense of a heart attack from the ECG. So we wanted to take away all this pain of the ECG, which takes approximately 8 to 10 minutes to finish, and undressing the patient to a very simple wearable device. We would just place this device on the chest and it's good to go. It gets connected to the Android and to the cloud and gets back with a fully cardiology reported event back to the doctors within about 3 minutes, and it will have clear instructions on what is the next step to be done. So this is what we would like to present. Our device is called Sidantech, and Sidan stands for heart in another language, Finnish. And we want to show you that we are very unique. We are the first 12-lead wearable patch. We are also multifunctional. So we can be used for doing a stat 12-lead ECG, which is like a 10-second record, or we can be used as a monitor, whether it's in the hospital, like in an ICU, or a monitor ambulatory at home. The same device can be used. Additionally, we are multifunctional. We have high resolution ECGs, which are very specific for increasing accuracy in various events, like identifying a heart attack or deciding on who is the best person to get which pacemaker. Additionally,
time up, Dr. Sugandhi.
Thank you.
Yeah, quick question from me. Of course, there's a lot of competition. Actually, there are already players who are doing this cardiac design labs, Nimble Heart, QT Medical, Sanket Life. But how do you propose to sort of compete with all these? In fact, there are others who are also sort of now working upwards from their 6 lead, 7, 5 lead machines to 12 lead machines. So how do you propose to sort of...
All of them are still using clumsy devices. Serial uses wires and the disposable electrodes. About a couple of billion electrodes are discarded every year because of this, of course, all over the world together. And they are not recyclable. We are completely green. We do not create any disposables. And we are wireless in acquisition and in transmission of the data, which nobody has done till now. The 12-lead ECG without wires is not obtained till now, and nobody is using active capacitance sensors, which are very difficult to tame. Additionally, we have also made it multifunctional. As I said, we are on cutting edge technology doing high frequency ECGs, which nobody is doing. And currently that scientists are only in a lab.
Okay. Thank you.
Doctor, this is Sai with Pfizer. So your presentation mentioned something about a peer review process. Can you talk a little more on what is that? Why is that necessary?
Okay, so currently our ECGs go to the cloud. They are analyzed by AI and an algorithm that is specific to us. And we would claim around 85 to 90% accuracy. In the past, even in universities, people have been working on it for 10 to 15 years, and they have only reached 85% accuracy. So we don't have a choice but to put some doctors there, also for us to accumulate enough data for sharpening our algorithms and the AI part of things. The conventional ECG machine just has some software that's put in at the time of manufacture, and people use it for 15 to 20 years without ever upgrading it. So all the results are very, very inaccurate. We would actually place the accuracy at 50% to 60%, not even in the 80s and 90s. So this needs a constant upgrade, and it needs a constant fine tuning to the hardware that is used, and that is what we are doing currently. So we have to have doctors to peer review it. So once we establish things and accuracy in the next 2 to 3 years, then it could be totally automated.
Thank you,
And
Dr.
commercially, you would, so one last question, sorry. Commercially, you would be looking at this as a service that you provide in addition to the device that someone has to pay for?
Correct, yes. So
Thank you.
pay per patient would be our model, at least for 2 to 3 years, so that we understand our device much better. And eventually it should reach the wellness market as well, and reach public service like kiosks where somebody can walk in with chest pain, get an ECG and get instructions on what needs to be done. So the process of going to hospital will be immensely shortened, and we can get the answer almost immediately and get them into the hospital as needed.
Thank you, Dr. Sugandhi. Thank you for your pitch. We'll call upon the next person, Mehak Malik.
Yeah, hi. Can you hear me?
Hi, Mehak. Yeah, can you see the slide?
Yes.
Okay. So, Mehak, you have 2 minutes. Your time starts now.
Yeah. Hi, everyone. I'm Mehak, and I'm building Cr Health. We're a digital health platform that provides sexual and reproductive health services to adults in India. It's 2020 and sex is still a taboo. Over 500 million individuals in India are in the sexually active age group. India is home to over 30 million STI infections and RTI infections annually. And there is an uptick in chronic disorders and lifestyle disorders that affect over 50 million women, that put them at the risk of infertility. The market for sexual health services in India is highly fragmented. It's run by providers who lack accountability, that ultimately leads to poor patient outcomes as well as satisfaction. Creating accountability and standardization in sexual health services will improve clinical outcomes in this market. We believe that empowering users with timely, accurate, and actionable information can improve preventive healthcare. And seeing that as an opportunity for disruption, we're using conversational AI to engage with young adults, enable them to ask questions in a discreet manner and get personalized, actionable advice. So our tools include a chatbot, which provides personalized risk assessments to users in a confidential manner, and holistic wellness programs, where we take a multidisciplinary treatment approach for chronic conditions by pairing patients with clinical experts ranging from gynecologists, psychologists to nutritionists, and help them live with their sexual and reproductive health disorders in a much better way. We're hoping with our work, we'll really enable Indian men and women to live their best lives.
Yeah, thank you, Mac.
So very quickly before other questions jump in, there are players like Menstrupedia, MZIL Education, as well as Econ. There are a lot of e-consult companies that are also doing something very similar. So how do you plan to sort of monetize this compared to all this?
Absolutely. I think that's a great question. So I think we are using content and chats as a way to establish our presence in the market and become sort of the first layer through which prospective patients come to our platform. But we monetize through consults and wellness programs. As an example, we're building our first wellness program, which is targeted towards women who have polycystic ovarian syndrome, which affects 1 in 5 women in India and is a leading cause of infertility, and also poses an increased risk for diabetes, thyroid, and cardiovascular conditions. So through a multidisciplinary treatment approach, by working with gynecologists, nutritionists, and fitness experts, they will be able to manage this disorder and be able to improve their or reverse their disorder. And likewise, there are other conditions that we want to build wellness programs around, including vaginismus, which is a disorder that leads to painful sex, sexual therapies for other sexual dysfunctions, and similar disorders for men as well.
Thank you.
Mehak Jaishree from NZR. So recently I've come across at least 5 to 10 different founders trying to work in the same space, right? What will be your USP? What will set you apart?
Absolutely. So I think there are a few emerging players in the market who are taking a product first approach where they want to introduce nutraceuticals and other good for you products as a starting point for patient journeys. We want to take a services first approach. We believe that that is an approach that will have better clinical outcomes. And I think the other USP for us would be we want to build something that is scalable. So our app would essentially enable sort of our target users to also track their behaviors as it relates to their conditions, which would include tracking activity, sticking to their treatments, and even conversations with their doctors all in one place, which is currently not there in any of the solutions that have just started to emerge. And I think this is a very nascent space so far.
Yeah. Thank you, Mehak.
Thank you.
Thank you, Jury. We'll go ahead with the next one. The next person is Dilip Shankar.
Yes.
Dilip, can you see the slide?
Yes, I can see.
Okay. So Dilip, you can start now.
Yes. Good morning, everyone. I'm Dilip Shankar, founder of Accord Innovation Private Limited. Let me start my presentation by asking you a very simple question. So why do people go for hospital? The obvious answer would be either to get treated or to get cured. What if I tell you 7 to 18% of the people who go for hospital tend to develop an additional problem during the course of hospital stay called hospital acquired acute kidney injury? And this is one of the problems which is commonly seen in ICUs up to 50% and 39% of post-major surgeries. So once patient developed this problem, they had to spend 3 to 7 days extra in ICU along with the associated expenses. And the current golden standard test, which is called as a serum creatinine, which is a late marker by 48 to 72 hours. Hence, clinician is not aware what is ongoing with the patient kidney function. And this problem also has a higher mortality rate up to 50% in adults, more than prostate cancer, breast cancer, and diabetes combined. And it also accounts to total $10 billion cost of the global healthcare system. Introducing a solution called AKI Care, AKI Care is a novel attachment to any traditional Foley's catheter system, providing a new diagnostic tool to monitor kidney function on real time by analyzing urine. AKI Care device mainly measure two major parameters. One, we measure accurate urine output and urinary oxygen tension to monitor the renal perfusion status. And this device seamlessly fits into an existing workflow where we are capable of providing a real-time risk assessment and early diagnosis in first 30 minutes using our algorithms compared to the traditional 48 to 72 hours. And we are highly affordable for hospital as well as for patients. So we at Accord Innovation Private Limited, hoping that we are going to provide a right amount of information to a clinician about the patient kidney function and prevent all avoidable death from acute kidney injury. Thank you.
This is Murli from Origin. What's the data generated so far? How does it compare to, does it correlate well with serum creatinine level?
Yes, sir. So as of now, we have done a small pilot study, and so serum creatinine is going to give us only 80% of sensitivity. However, in terms of evidences, the combination of urine output and urinary oxygen tension can provide 94% of specificity and 93% of sensitivity. And it is going to be, and there is a lot of evidences which are already available in terms of research. Also, some of the people have done in terms of a validation on humans as well as on animal, and it is giving a very positive data.
Hi, this is Nitin here. There are, of course, several, almost 15 biomarkers that have been tested and validated for the same problem. And of course, there's an FDA-approved device that's already in the market, which also looks at the growth factor. So how do you think this particular ratio that you're looking at will compare with some of these, especially the very quick checks in terms of the biomarkers, especially, I should say?
Sir, in India, we have only NephroCheck or we have NGAL, which is taking at least 6 to 8 hours or more than 12 hours. However, in other countries, there are NephroCheck, which is a biomarker-based test, and each test is more than 6,000 rupees per patient. For example, if a patient who is undergoing cardiac surgery is on risk of AKIs, every 2 hours they're going to test. And if you just rupees for every 2 hours, it is going to add up a lot of device. For example, our disposable is just 200 rupees where we can monitor the kidney function on real time for 24 to 48 hours.
Okay, thank you.
So do you have any plans to look at other markers beyond what you have just started with?
Yes, sir. So as of now, we are looking for urine output and urinary oxygen tension for the risk assessment of acute kidney injury. And we have a very unique feature where we can automatically extract the urine sample from from the urine bag, and we can analyze any of the urinary biomarkers at the bedside. So we already have developed a marker for urinary dipstick where we are currently getting like 14 different parameters, and we are currently modeling different kidney function modelings, also for the disease progression.
Okay, thank you.
Thanks a lot.
Thank you, Jury. Thank you, Dilip. We'll go ahead with the next person. I would like to request Dr. Sanjeev Sambandhan.
Hello, ma'am. Can you hear me?
Yes, Dr. Sanjeev. You can see the slide?
Yes, I can. Thank you.
Okay.
So firstly, good morning to all of you. And thank you for giving me this time to present my little idea, which is something called as an edible electronic pill for diagnostics. So if you look at the medical diagnostics state of art at the moment, what you have is you have people going to clinics or doctors or going to an expert to get some very reliable diagnostics performed, which is typically invasive. And the reason why invasive diagnostics gives you reliable results is because your analyte concentrations inside the body are much higher. But at the same time, it uses up the time and energy of the expert, which might be a doctor or some supervisor in some remote area. On the other hand, you have non-invasive technologies, which is like your wearable electronics, which need not always give you very accurate and timely results, but it doesn't need any supervision. So you save on the time and energy, which is a very precious resource. So can we marry these two? Okay, so that's the key idea here. And the idea is to see whether we can make invasive diagnostics not require supervision. And in order to do that, you should make sure that the invasive diagnostic process, which will give you reliable results, is both safe to use as well as easy to use. And therefore, this is the problem that we are trying to solve. Now, our solution is to come up with a sugar pill, essentially, which is building electronics on sugar as a substrate. and it's all packaged into a little standard capsule, and this capsule is swallowed by the user. And once it is swallowed, the capsule goes through the gastrointestinal tract and is triggered at some appropriate point, and it performs diagnosis of analytic or clinically important parameters. It senses different analytes, which could be pH or bleeding in the gut, etc. And after it performs this diagnosis, it radios this message to the outside world. It's in fact radioing the message to a little patch worn by the user outside. It's simply got a coil on it. And after that, the pill is safe and it's digested by the body. So
yeah,
the
time
contents,
up.
yeah. So that's my little idea. Yeah, thank you.
Hi Sanjeev, this is Taslim Ayub here.
Yes,
so where does this idea stand right now?
So right now we have all the engineering in place, so which means that we know how to build an energy source out of sugar. We basically use galvanic cells. We know how to do communication. We have got LC oscillators that will communicate at RF. So all the engineering is done. What we have not touched at all is the clinical trials, which is we now need to take this platform that we have and see whether it can actually perform the whatever was expected inside the body. So we have not started clinical trials at all, but what we have seen is an engineering platform that is reasonably reliable.
When you say clinical trials, you possibly would do animal trials possibly first.
Animal trials, yes. So we will first try it out for, we will first try out in vitro trials for certain specific cases. So if for example, what I imply is, let's say we imply bleeding in the gut. We want to see specificity. pH is easy. pH is something that we can easily do, but we want to be very specific to certain proteins that we want to sense, etc. So once we are confident about that, then we start doing animal trials and human trials. But yeah, so that entire segment is left unattended to.
And this is a very quick one. I think yes or no would suffice. This is in a form of an individual right now or in a company form?
Right now, it's an individual.
Okay,
it's research from my lab. Yeah.
Thank you.
Sanjeev, this is Ashish from Novozymes.
Sorry, Ashish, you broke off.
Among the analytes you mentioned, which would you prioritize the high?
Yeah.
Sorry,
so for
I'm
this,
saying that among the analytes you have in mind, which are the top analytes?
Yeah. So we approached a lot of. Thanks. Yeah, I got it. We approached a lot of GI specialists for this, and we listed out what they feel is useful information for them. And the top three in the list are, one is pH, so a spatial variation in the pH for people with GERD, for example. The second is a microbiome analysis of stool samples, and the third is bleeding in the gut. So these were the three parameters, or clinically important parameters that the GI specialist felt would be very helpful.
And so from a practical standpoint, do you expect the patients to take just one pill, or there'll be multiple pills with different detection systems, or how would that happen? Because it's going through the entire gut system at various spaces. So how do you manage to collect all this data?
Good question. So for each analyte right now, the plan is to have a separate pill. Okay, now it could be merged. But there needs to be certain engineering problems that need to be solved. Now, for example, if it's pH that you want to sense, basically I would say two orthogonal parameters can be merged together in a pill. So if it's say pH and detection of some other protein, that is perfectly fine as long as one measurement does not interfere with the other. So that's as long as you have orthogonal parameters wherein one measurement or one parameter does not influence or impact the other one too strongly in terms of sensing, and I'm talking about the sensor, we can package as many sensors onto this one pill as possible.
Sanjeev Jaishree from India. Why would this be better than a combination of endoscopy and the other routine tests that are done today?
Yeah, again, for a great question. So we already have things like capsule endoscopy, which is let's say like a MEMS device, which has got cameras and swallow a pill. But the biggest USP, the biggest advantage here is you do not need a specialist. Now, let me give you a scenario. Let's say there is a remote area and people are swallowing this once a week, let's say. And what you can do is a doctor sitting far off can now, without interacting with that community, get a statistics or get a feel for what's happening there because of these measurements. So it does not require supervision and it is safe to use. So that is the key advantage you have as opposed to going to a specialist and getting an endoscopy, etc.
Thank you.
Thank you.
Thanks.
Thank you, Mr. Sanjeev.
Thank you, ma'am. Yeah.
Thank you, jury. I would like to go ahead with the next participant, and I would request Dr. Ravi Adgulwar. Can you see the screen?
Yeah,
can you see your slide?
Yeah. Am I audible? Yeah.
Yes, you're audible. So Ravi, you can start.
Yeah. Good afternoon, everyone. I am Ravi Adgulwar, co-founder and CEO of Avisa Myco. Today with me is my co-founder, Dr. Ravi Kumar. We have developed a technology to produce melanin from a proprietary strain of fungus. Melanin is this pigment in our skin and hair which gives its color and protects us from ultraviolet radiation. Melanin is evolution's answer to radiation. Melanin is a multifunctional supermaterial with amazing industrial properties. It can block UVA, UVB, and UVC, and hence can be used as a UV filter in sunscreen. It can block X-rays and gamma rays, and hence can be used as a shielding against radiation in aerospace, defense, and medical applications. And it can have many other applications. But currently, melanin is not used anywhere because it is costly. It is sold at $400 per gram, that is 8 times costlier than gold. And our technology solves this affordability problem. We can produce melanin at 10 cents per gram, that is 4,000 times cheaper, which opens up many new industrial opportunities which were earlier commercially not viable. Currently, we are focusing on cosmetics, especially sunscreen applications, because the current chemicals and nanominerals used as UV filters have problems. The chemicals in the sunscreen are damaging the coral reefs and the marine life they sustain. Many of the beaches in Hawaii and Florida have banned chemical sunscreen. Cosmetic industry is looking for an eco-friendly solution. The other problem is the chemicals and the nanominerals seep through the skin and get accumulated in the body and are not disposed of, and which causes many side effects. US FDA has ordered a safety review of 8 major sunscreen ingredients, which safety review is currently on hold under law pressure. We believe our melanin can be the solution to the current problems faced by the UV blocking industry, as melanin is biodegradable, safe, and effective UV blocking solutions. We plan to file a new drug application, NDA application with US FDA, as sunscreen is considered a OTC drug in US. We have raised our pre-seed round and we are in the process of currently raising our seed round. Thank you.
Dr. Ravi, this is Sai with Pfizer. So your the cost that you have projected in your presentation, is that your actual or that's what you expect it to be once you scale it up?
Yeah, we have scaled it up to 100-liter bioreactor stage, and this is the cost validated at 100-liter stage, that is 10 cents per gram, but we expect to bring it down to below 1 cent per gram. Eventual target.
Ravi, this is Ashish from Nova. Ravi, this is Ashish from Novozymes.
Yeah.
Normally, microbes make these metabolites as a mixture of analogs and congeners. Is your product a complete clean chemistry, meaning a single compound?
Yes. We are getting around 99% purity melanin.
Okay.
Hello, this is Murli. How does the production of melanin with your method?
My second question was that in your...
Hello. Yeah, come again. Hello.
Yes, Dr. Ashish, you can go ahead with the second question and then we'll take the next one.
Okay, I'm sorry. My second question was that in your video, you mentioned that that your target is to get to 95% UV blocking, right?
Yeah.
And currently you are at
66%. In the formulation, so melanin can block in the mixture
with already 4% of the
for SPF 4.
Right.
SPF
My question
4.
was that going from 4% melanin in SPF 4, 66% blocking. Now you want to get to 95% blocking.
Yeah.
And you're already adding 4% of your active.
Yeah.
How high can you go in your formulation? Yeah,
4%.
As such,
You see, as such, toxicity wise, there is no upper limit. So we expect we should get a SPF of 30 with around 8% melanin. That is our expectation, but actual results are yet not there. It is a work in progress.
Okay, thank you.
Yeah, so this is Murali. My question is, there are number of other bacteria also known to produce melanin, bacteria or other microbes.
Yeah.
How does your method compare or differ from the other reported systems?
You see, every living organism produces melanin. But the problem is the melanin is produced in very small quantities, and that too in a cell wall, and some bacteria extracellularly. The USP of our system is it produces in huge quantities, which is unreported. We are the only company which is having this commercial melanin technology. So for melanin technology, we do not have any competition, and we can produce it at a very cheap cost. So that is our USP.
Yeah, hi, Ravi. This is Suman here from L'Oreal. You mentioned about US FDA approvals. Actually, can you just comment on the timeline?
Can you repeat? Can you repeat? Your voice is not clear.
In terms of US FDA approvals, can you comment on the timelines that are required to register as filter?
I could not get your question. Your voice is breaking down.
Is this clear?
No.
Okay. I'll skip this one actually. Sorry.
Okay. Yeah, we can.
So how is your technology IP protected, Dr. Ravi?
We have filed a patent in India and it has been granted. Over and above that, now that patent in India is for process and production. And so now additional patent we have filed in US for around applications of melanin. But we plan to file additional 5 to 6 patent once we complete our seed round.
And is your organism genetically modified or is it as naturally occurring?
Right now it is naturally offering, but we plant, so we are in the process of modifying this organism to increase the productivity.
Thank you.
Thanks. Thank you very much.
Dr. Osman, do you want to try asking the question?
Is this clear now?
Yeah, it's better. Yeah, it's better.
Okay.
You can ask. Yeah.
Yeah, Ravi, you just mentioned about FDA approvals and those things. I just want to understand the timelines that is required to get registered as filters. Do you have any idea about those?
Come again?
FDA approvals to register as a filter. Do you have any idea in terms of timelines?
Timelines, actually we have one consultant in US who is advising us on the FDI approval process. He is also our technical advisors, and based on his input, we have been projected expense of around $500,000 and around 2 to 3 years.
Okay.
Thank you, Ravi. Thank you for your pitch. We'll go ahead with the next participant. I would like to request Dr. Srisha Malayal.
Hi.
Hi. Yes, you are. And can you see the slide?
Yes, I can.
Okay. So you have 2 minutes for your pitch.
Yeah. Hi. Hi, I'm Dr. Srisha Malayal, co-founder of Bio and Sustainable Solution, and I hold a PhD from Indian Institute of Science. The biggest environmental concern that we are facing today is the disposal, treatment, and pollution caused by single-use plastic. And that is the problem we are addressing today. Very few alternatives are there in the market today for single-use plastics, and these require minimum 6 months to degrade in the environment, and also in the process release microplastic-like substances and chemicals to the environment. Other alternatives that exist is a polymer made from food-grade starch, again, which pose a direct threat to food security. Our solution is a 100% biodegradable alternative to single-use plastic, which degrades in less than 60 days. This material is made from natural plant fibers and latex. The natural fibers are obtained from various agro waste, by fermenting them in an innovative biogas plant where the agro waste is fed as it is without pulverization. Therefore, by controlling the fermentation time, it is possible to extract natural fibers as maybe long as 2 meters in case of banana stem, along with biogas and fertilizer. The fibers obtained in this manner is heat pressed at 120 to 150 degrees, wherein the fiber forms a mat-like structure and which is again sprayed with latex to make it water resistant and flexible. So our value proposition is 100% biodegradable alternative for single-use plastic, which composts in less than 60 days and which is at least 3 times cheaper than any other product in the market. This is because the natural fibers are a byproduct of fermenting agro waste, which makes the cost of substrate and processing zero. Since the process of natural fibers occurs at farm level, this process ensures additional income to farmers through sale of natural fibers and also access to clean energy and fertilizer at very low cost. This also creates circular economy from waste and also ensures the triple bottom line of sustainability. Thank you.
Yeah, one Paresh Vora. Can it be used for this rice husk or whatever that is a big problem in Punjab and all in the winter?
So we have not done from rice husk, but we have done from paddy straw.
Isn't that same? Isn't paddy and rice same? Oh, not husk, sorry, the straw. I meant the straw, sorry. I used wrong.
Not from the husk. The husk fibers are too minute.
Yeah, basically straw is a problem, right?
Yes, we have done from straw, we have done from banana stem, sisal, hemp, flax, and quite a lot of other substrates.
Ashwin here, what is the production per kg or per acre, whatever the waste you get, how much fiber you can produce from that? And second question to that is, how are you competing with the cost? Because these technologies are good, but then the cost of production is way, way higher. So how your technology is answering that?
Okay, so your first question. So each ton of agro residue processed, for 1 ton, you will get approximately 100 to 150 kgs of fiber. So this fiber, we are mixing in a 2:1 ratio with latex. And we can, so for each ton of agro waste produced, you can make at least 9,000 single-use plastic bags of the size 16 inch by 16 inch. Okay. And the next question of yours was on the cost. See, so here the substrate is agro waste, which is again an issue and people are burning it. So there's no cost for the agro residue and the processing happens using a modified biogas plant, which again, what happens is the fiber is just a byproduct of a biogas plant. The main products are biogas and fertilizer. So there is no cost involved in the substrate and processing, which gives us a upper hand in making it at least 3 times cheaper than any other existing product in the market.
Srisha, this is Sai from Pfizer again. If you look at this as a production rather than waste handling technology, the volumes that you need to process would be far higher than what probably you are able to handle now. And if you look at that supply chain, I would suggest maybe you can do the economics for collection and transportation, etc., and processing of that waste, and truly see whether your cost is competitive with what is available in the market today.
Right. So as of now, we have kept at least like 10 rupees for the farmers, wherein we will buy back at 10 rupees per kg of fiber from the farmers, which is a cost which we have decided, which I think may include the transportation cost and all of it. Keeping this 10 rupees per kg, we have estimated that we can still make a 16 inch by 16 inch bag at 1.6 rupees, which also includes a 20% margin.
Yeah, this is Ashish from Novozymes. Building on the same question, that the real impact of this will come if you can really scale it up, right, because you have aspirations to supply to companies like Amazon. My question was on the quality. Depending on the feedstock and the fiber length and composition, your quality might change, right? How would you address that?
So we have done. So I basically started this with my PhD from IISc. So we have tested all these possibilities like the tensile strength, the material, almost most of the fibers that we are tested, we are getting an average of a good quality or the similar quality we have got from almost all the substrates we have tested.
Hi, this is Karthik from Sangam. Could you just speak about the novelty of the biodigester itself? I'm assuming there's obviously specific feedstock you need. right, with like some level of cellulosic material.
Right.
But what does an existing biogas plant be able to use it, or do you need a whole new different type of digester to be installed? And can you explain the technology a little bit more?
Sure, sir. So we can't use an existing biogas plant because all the biogas plant that exist in the world today is based on the principle that you have to pulverize or make a slurry out of the input material. You have to add water, you have to grind it and then feed it into the biogas plant. In such cases, you won't be able to extract fibers as this length. That is, I'm talking about at least 2 meters from banana stem and all that. So our biogas plant is a specific or an innovative one, and we have to install this particular biogas plant to extract fibers. Here in this biogas plant, we are feeding the material. So if you talk about a banana stem, which is almost like say 2 to 3 meters, the length, we are feeding as it is into the biogas plant, allowing the fermentation to occur. Basically, I'm controlling the fermentation time or the retention time in the reactor and trying to get the natural fiber. So for each type of residue, the retention time is different. For banana stem, I have a optimal retention time of 24 days. Whereas if I take areca husk, it is around 30 to 35 days.
Yeah, Dr. Srisha, the time is up. You can keep your answers brief.
Okay, just last question. What can you do a 50 to 100 ton day plant?
Sure, sir. So we have already operating a 1.5 to 2 ton processing same biogas plant with different substrates. It's just that we are doing it on agro residues, but without fiber recovery. So we have this design already in the market for various other substrates. We're just modifying it to accommodate the fiber recovery.
Okay. Okay.
Thank you, Dr. Srisha.
Thank you.
Thank you, jury.
Thank you.
We have the last pitch for today, and I would like to request Prerna Tonke to present. And you can see this slide, right, Prerna?
Yes, ma'am.
Okay. So, Prerna, your time starts now.
Okay. If you believe there's a magic on this planet, yes, it is definitely present in the water. Clean drinking water is the secret of healthy life. Hello, everyone. I'm Dr. Prerna Tonke presenting NanoPearl, a step towards clean water. Access to clean drinking water is becoming a major challenge all over the world. With the 1 death per 90th second, India is running towards its next epidemic just because of the waterborne diseases due to the 3 major contaminants present in the water: TDS, heavy metals, and the microbes. Instead of having a multi-million-dollar water purifiers market in India with the CGR of the 22%, only 3% of the India's population uses the water purifier. This is mainly because unaffordability, lack of awareness, and most important, we do not have a single product in the market that can remove all these 3 contaminants in the single product. Thus, we at Dew Technology have developed a solution called NanoPearl, which is a multivalent 2-stage water purification system. In the first stage, we have utilized nano-engineered hybrid crystal structure, specially made up of aluminum and polymeric unit to remove the TDS. And in the second stage, we have utilized copper and zinc unit, which is the hybrid crystal structure with the absorption capacity 3000 mg per gram that possibly allows absorption of the heavy metals, and walls of the copper allows a antimicrobial capacity. Currently, we are impacting around 7,000 lives in all over the Maharashtra by implementing this technology into the community filters in some schools and colleges as well. We are also working on some of the water awareness camps. This is an affordable and scalable technology that costs around 1,000 rupees per unit per household purifier with the margin of 80%. Recently, we have been approached by some of the real estate business and the beverages industries for more than 1,000 units till mid 2020. The electricity requirement is optional here. If the trends continue, soon we will become a multi-billion-dollar company. So let's work together for clean, green, and healthy future. Thank you.
Paresh Vora here. Can I just quick question? What is the highest TDS you can handle?
Sir, we have tested it for the 1,000 ppm TDS, and then it comes down to the 300, means 200 to 300 in the range.
Okay. What about like 2,000, 2,500? Do you think it can handle?
Yes, it can handle it, but probably probability like whatever the analysis we have done throughout the India, we got to know that 1,000 ppm is the maximum TDS that we can.
No, not well. That's not true at all. In Gujarat, it's very common to have 1500 plus and even 3000, 3500.
Yes, sir. It's a life of the purifier is the 4000 liter, and if we allow for it's like it can reduce the life, but we can recycle it for the 4 times. So if the life is get reduced, we can recycle it again and we can reuse it.
So Prerna, is your crystals are they recoverable or are they one single use?
Also, these are the recyclable. Like 4 times we can recycle it. We just need a back pressure wash with the hot water, and we can keep it into the sunlight to dry, and we can reuse it again.
4 times, you say?
Yes, sir.
Okay, and I'll echo the other gentleman who spoke, right? In my house, the TDS is around 1500 to 2000 right now. So I think you should think about that.
Yes, sir.
Priya, this is Ashish from Novozymes. You mentioned using an organic framework.
Yes, sir.
Have you tested that no other components are kind of leaching out into the water
Sir, actually,
over time?
Yes. So actually, these are the frameworks, like these are the cage-like materials where after the formation of the nanopell unit. So these are the walls made up of the zinc and copper unit, and also the aluminum and the polymeric unit. So they cannot leach out the compound. It's a cage that is formed with the these are the metal organic frameworks which are very strong bonded and that cannot be leached out in the water, because we are not using any adsorption of other materials on it. We are making a cage out of it.
Okay, thank you.
Can you do a community level plant, like a village level plant, which maybe 1000 liter per day or something?
Yes, sir. Right now we are already implemented it into the Parbhani district in some of the villages where there are 200 to 400 people are living in one community. We are using it on the community as a community filter. So hopefully we will be scale up it for more volume as well.
And what's the net cost per liter of water? Have you made an estimate for the user?
Sir, for 23 liter household purifier, it cost around 1000 rupees. So as it is, it can be for family of a 4 person. We can use it for 3 months.
Per liter of water, is there any estimate of cost per liter or per kiloliter or something?
No, sir. For per liter, we have not calculated. As if we are working on the water purifier for the like a household purifier. So we have calculated for that.
Yeah, that's fine.
Yes.
Thank you, Prerna. Jury, do we have any more questions? Okay, looks good. Thank you, Prerna. So we have completed all our pitching for today, and thank you all the participants for being here. And I would like to request Nandita to take over.
Yeah, thanks, Debolina. I think we did it in reasonably good time. We were, I know, worried that we would go on and how we would manage this. We had also considered actually outsourcing this whole, process to one of the event managers. But I think the team took a good call to do it ourselves and manage it ourselves. So I hope it's gone well so far. Also, if there were any hitches, apologize for that. I think we've come to the moment now where we really have to come, eagerness to find who the winners are. At this point, I would request the jury to, we need to move to the other jury discussion room. You would have also got the link on your cell phones. I think we have sent the other link. You may choose to join that link through your cell phone and stay connected on this so that you don't need to sign in, but I leave it to you. We could possibly move to the other links. So I request all the jury members to use link 2 and join the jury discussion meeting, and then we will come back here. In the meanwhile, I think for the participants, maybe we can show them a short video about the journey of NBEC so far. Also, we are very happy that NBEC has really penetrated into the hinterlands of our country. So we will also take a short glimpse of the participants from the tier 2 and tier 3 cities, of which we are so very delighted to have them. Having said that, I will also go to the jury discussion and request my colleague Debulina to take this forward from here. And all the jury, we will come back here. Sorry, Debulina. Just okay. That we will come back here to this link. Please, let's join the other link now. Thank you.
We can exit this link right now, right? Just quick question. Jury can exit this link and then rejoin, right?
Yes,
yeah, you can exit and rejoin also.
Okay,
yes. Okay. Yeah. Can do that.
Ms. Nandita, just a quick thing, right? Since we've been here since 9:30 or so, can we have a short break before we get into the jury link?
We can take a short break. Maybe for both sessions, we can take a break, Debulina, now. And maybe show the videos after about 10 minutes.
Sure.
Yeah, thanks so much for that.
We can just
so what time do you want the jury to join, Nandita?
So Nandita will be joining with you, with all the jury members.
Maybe we can join the jury discussion at about 10 to 12.
Okay, we'll do.
11:40 now,
that's
so 11:50, 50.
Sure.
And then come back here after we are done.
Sure. Thank you.
Yeah, in case you're joining on, continuing to sign in here, I would just request you to put your be on mute.
Sure.
For obvious reasons.
Thank you.
So I would like to request everyone, we can take a break and we'll be back by 11:50.
Sure. Thank you, Debulina.
Sure.
अभी फिर वही दिखाएंगे कैसे हाउ
Hello, everyone. I hope everybody is back from the break. Hello, everyone. I hope everybody has come back from the break. And we are going to play a short video of NBEC journey. Good afternoon, Srimati Anju Bhalla. We welcome you here today.
Good afternoon.
This is Debolina from CCAMP, and we have we have Bhavisha Debarshini Chandrika from CCAMP and Dr. Niranjan also. So I was just telling the participants that we are going to share a short video on NBEC journey. So I'll just try sharing the screen and we all can have a look at that.
Have I joined too early?
No, it's fine. It's perfectly fine. We are glad that you've joined.
Okay.
So, Srimati Bhalla, it is like jury has gone to the jury discussion room. All the pitches are over. And they have gone to the jury discussion room to decide on winners. So by the time we watch videos, probably they should be back. Okay.
Okay. Otherwise, if I wanted to log off and rejoin, what would be a good time?
12:30 would be a good time, ma'am.
Okay.
That would be good. Yes.
I think I'll do that then.
Sure.
Okay.
Sure. Thank you, ma'am.
Thank you.
Bhavisha, you can see the screen, right?
Yes, you can start. Debolina, we cannot hear the video. Audio.
Sorry, I'll just try again.
Yes.
for previous years, you all have had such a wonderful response that I'm confident that this year you're going to go much, much higher than what it was earlier. So heartiest congratulations to all of you. And it's a real pleasure to officially launch this NBEC. And if these enablers have helped us to reach where we are, today our responsibility is not to move up the ladder one step at a time. I think now it's our responsibility, as we always say, to leapfrog and to be really up there, because that's what India is now, we feel absolutely ready for.
It's always such a pleasure to participate in launching this year's NBEC, like it's always been every year. And I've always believed that, translating science into business through entrepreneurship is the biggest testimony to what we can do with exciting science.
Particularly the feedback received on the pitches has tremendously helped us to transform our pitch decks and improve our value proposition.
How can I pitch the impact we are creating to the people around? And it has actually developed some pitching skills in me.
Scholar at IIT Gandhinagar. My team is developing virtual reality-based hand rehabilitation and assessment system for stroke patients.
Student of Biotechnology, Basveshwar Engineering College, Bagalkot, Karnataka. We are making an ethno-veterinary product against animal ticks using biodiesel industrial byproduct.
An early startup based at Patiala, Punjab. We have a novel intervention called Gola Fresh to reduce the post-harvest losses in fruits and vegetables.
Thank you, everyone, to be a part of our journey in this NBEC 2020. So next, I would like everybody to watch a short video. As Nandita had just said, NBEC has not only penetrated to metros, but to many other cities and towns all across India. And so what we did, we got some snippets from our participants from across tier 2 and tier 3 cities. And we have made a short video. So I would like to request Dr. Bhavisha to share the video with all of you.
Hello everyone, my name is Chandan Kumar Jha and I am a PhD scholar at IIT Gandhinagar. My team is developing virtual reality based hand rehabilitation and assessment system for stroke patients. The objective of our product is to make the rehabilitation therapies engaging and accountable by means of virtual reality games. The entire BREC program has been a very phenomenal experience for us, particularly the feedback received on the pitches has tremendously helped us to transform our pitch decks and improve our value proposition. And therefore, we would like to thank the entire BREC team for making this event successful and helpful for us. Thank you.
Hello everyone, this is Kalyani Shinde, founder and managing director of Godam Innovations Private Limited. We work into agri-tech sector. We specifically work into onion ecosystem. We help farmers reduce post-harvest wastage. We are working towards redefining smart warehousing. We are based out of Nashik in Maharashtra. Working with NBEC in the previous rounds have really helped me to understand how can I pitch the impact we are creating to the people around, and it has actually developed some pitching skills in me, and also how the numbers look like. Other than that, the learnings have really been good because the inputs which have come from the jury is actually going to help me in the future for scaling the business and for increasing the impact we are creating for the farmers. So thank you so much.
My name is Sanjay Saxena and I'm the founder and CEO of AgFarm Bioinnovations, an early startup based at Patiala, Punjab. We have a novel intervention called VolaFresh to reduce the post-harvest losses in fruits and vegetables. The product is cost-effective, eco-friendly, energy neutral, and non-residual. Apart from it is eco-friendly, it is user-friendly, and it enhances the shelf life of the fruits and vegetables by 7 to 21 days on an average. The guidance of the speakers and the mentors in the NBEC bootcamp have been of immense help. However, the duration in one-to-one session was too short. So the one-to-one session of the selected pre-final startup should be for a longer durations with detailed deliberations. The overall experience and participation in the NBEC 2020 was a very good and exhilarating experience. Thank you.
Hi, we are Raman and Lakshman from Backyard Creators. We are a startup building an innovative tech to solve medical, social and environmental problems. Mainly we are working on adhesive non-surgical hearing aid for by birth and totally hearing loss patients. What problem we are going to solve is there is no more implantation surgery needed for the hearing loss. And we have developed our knowledge through the CCAM bootcamp. The CCAM bootcamp is very helpful for us and we have a clear view about after the mentoring section about our business model. Mainly we are very well known about our business model now. Thank you for that CCAM.
Hello everyone, I am Nikita Chingalgi, student of biotechnology, Cr Engineering College, Bagalkot, Karnataka. We are making an ethnoveterinary product against animal ticks using biodesign industrial byproduct. It was a great opportunity to participate in National Bioentrepreneurship competition. It was so encouraging for us to understand and learn more things through the team of NBEC. The sessions were helpful for our pitch in more efficient, effective and even to make more competitive. Though mentors made it still easier by guiding us in the right direction after every presentation. This made us more confident to start entrepreneurial journey. Thank you CCAM team for making it easy, informative and giving us this opportunity.
I'm Ekta Kishore Gompulwar from MITWPU Pune. Our leader is Shraddha Khobrakade. Our domain is antimicrobial resistance. We are solving the problem of antimicrobial resistance on the catheters by coating the catheters by the polymer and the herbal essential oils. By performing in NBEC camp, it is a great experience and there are a lot of learning things we can get from it. That is presentation skills and how to enhance our research work and how to study about the future goals and the market value of our product. Thank you.
Thank you, everyone. I would like Dr. Niranjan to take over.
Niranjan, you are on mute.
Thanks for telling that, Bhavisha. I think it's been a grueling session for especially the grand finale finalists. We will now take a short break. Our next session starts at 12:30. I'm sure all of us are waiting for the session, so I request all of you to reconvene at 12:30. You can be logged into this link, but we will reconvene at 12:30.
Good afternoon, Dr. Devan.
Hello, very good afternoon.
This is Debolina. Hi.
Hello, Deblina. Hi, how are you?
I'm good. How are you?
Good, good, good.
Actually, the jury discussion panel is going on in another link. So we are now waiting for them to join back so that we can have your address and the winner announcements.
Sure. So it seems there is some time. Should I log back again in maybe another
No, I
5-10
think
minutes?
5-10 minutes. Yeah.
All right. Okay, sure.
Thank you all for rejoining. I think the grand jury is still in discussion to choose to pick up the winners. We will wait for a few more minutes. I'm requesting all you to continue logging in and stay tuned. I just received message from the grand jury room. There is a fierce discussion ongoing with respect to picking up the winners. It will take about 10 minutes, 10 more minutes, requesting all of you to please continue logged into this meeting room and let's wait for our jury to reconvene. Thank you all for your patience. A grand jury is still in discussion, and the winners are being picked. It is being discussed with grand jury is in another meeting room. In the meantime, I'm requesting my colleague Debolina to play a short video clip on the journey of the National Bio-Entrepreneurship Competition. We had played this video clip just about half an hour ago, but not all of you were present at that time. So I'm requesting my colleague to play the video clip again. Debolina?
Yeah, sure, Niranjan. I will take a few minutes to start the video.
Sure.
previous years you all have had such a wonderful response that I'm confident that this year you're going to go much, much higher than what it was earlier. So heartiest congratulations to all of you. And it's a real pleasure to officially launch this NBEC. And if these enablers have helped us to reach where we are, today our responsibility is not to move up the ladder one step at a time. I think now it's our responsibility, as we always say, to leapfrog and to be really up there, because that's what India is now, we feel absolutely ready for.
It's always such a pleasure to participate in launching this year's NBEC, like it's always been every year. And I've always believed that, translating science into business through entrepreneurship is the biggest testimony to what we can do with exciting science.
Particularly the feedback received on the pitches has tremendously helped us to transform our pitch decks and improve our value proposition.
How can I pitch the impact we are creating to the people around? And it has actually developed some pitching skills in me.
I'm a PhD scholar at IIT Gandhinagar. My team is developing virtual reality-based rehabilitation and assessment system for stroke patients.
Student of biotechnology, Basveshwar Engineering College, Bagalkot, Karnataka. We are making an ethnoveterinary product against animal ticks using biodiesel industrial byproduct.
An early startup based at Patiala, Punjab. We have a novel intervention called Gola Fresh to reduce the post-harvest losses in fruits and vegetables.
Thank you, Debolina. I understand that the grand jury had just finished their discussion, and they are shortly joining this room back again. Let's just give a couple of more minutes, few more minutes, and then we will start. Welcome back to all of you. This is the session that we all have been waiting for, the National Bio Entrepreneurship Competition, the grand finale, final session. The NBEC, as we call it, has gone around the country over last 4 months. There have been passionate ideas that have been pitched. The jury have asked some difficult questions. There have been passionate counter arguments, and this has happened over multiple rounds. Today is the culmination of that process. As one of the grand jury members very aptly said earlier this morning, that reward is the journey. And the final is all of you. You have gone through this NBEC journey over the last 4 months, and I hope that this experience has been rewarding. Nonetheless, this is a competition and there must be winners. And today, this session is when we will announce them. Well, we know the winners, but you will have to wait for just few more minutes before we make those announcements. I am now inviting Dr. Taslim Arif Sayed to welcome this August gathering. Dr. Taslim Arif Sayed is the CEO and Director of Center for Cellular and Molecular Platforms, CCAM, India's premier bio-innovation hub. A neuroscientist by training, Dr. Taslim is a strong proponent for deep science-led innovation and research translation in the Indian bio-innovation ecosystem. PhD from Max Planck Institute, Germany, and trained in management for biotech innovation at QB3 at University of California, Santa Cruz, UC Berkeley and UC San Francisco, USA. Dr. Taslim has been spearheading CCAM's efforts and building of this biotech innovation ecosystem through CCAM over the last decade. I'm now inviting Dr. Taslim Arif Sayed to welcome the gathering.
Thank you so much, and good afternoon, everyone. And participants from the US, a very good evening. Honorable Deputy Chief Minister who was to join, couldn't join for something. Dr. Swaroop is joining through the video message. Dr. Kiran is joining through the video message. We have Dr. Bhala and Dr. Devan who would also be here. So warm welcome to everyone and the other dignitaries. It's an absolute pleasure to get into the final session of the NBEC 2020. I'll just try to share a very few quick things in terms of the effort largely that we are putting it together. So as you know, CCAM is based out of Bangalore, but through its efforts, we have innovations and presence across India. And the broader and the larger mandate of CCAM has been to foster deep science research and innovation. For last 9 to 10 years, we have been doing it. And as we grow forward, as we build further, as we take more innovations to society, I think what is meaningful is that the people are being served through that. And to be able to achieve it, we'll have to work on the ground. And NBEC is one of the classical example of working on the ground and unearthing the talent of India's bio entrepreneurship, from any state and every union territory of India. As we know, and you just heard in the clip from Kiran, Dr. Kiran Majumdar, that science entrepreneurship is actually a phenomenal vehicle to take scientific knowledge to the next. And scientific knowledge reaches its maximum when it actually solves a societal problem. I think we all clearly know that, and I'll not say much now. We have many examples, as recent examples, 2 quick examples that we talk about today. The brilliant technology in terms of the mRNA-based vaccine is a brilliant scientific understanding that people have been able to build. And the first 2 approved the vaccines are both based on the mRNA delivery technique. So it's clearly shows that how new science will constantly change the way we will be able to help the society around us. And what we, all of us as scientists and the innovators want to do is to make sure that we can build new capabilities. What we heard through the morning today is actually very, very exciting. We heard many, many new ideas which would represent the way going forward, that those were the new innovations which will serve the society. And India, we are seeing completely change in landscape in bioinnovations. A lot has actually come from the BREC, the establishment of BREC and BREC support, which is actually a funding partner as well for the NBEC and the BREC per se. This has been a broad impact of CCAM as in the entrepreneurship vertical. While we have a research technology platforms are different, but what you see is that the ability to constantly engage with innovations and do it, and then have a few of the products already serving people has been very, very important. So this is what you call it investment, not financially, investment in deep science innovations. And when are those, I just wanted to share when those innovations become actually valuable, right here. In around late March, early April, we launched CCAM COVID-19 Innovation Accelerator, which was an attempt to answer the unavailability of healthcare solutions for India for COVID-19. We didn't have much coming our way, no import, no availability of other testings, ventilators, and so on. And we, along with many partners, actually launched this accelerator to identify innovations right in our backyard and see if those are ready to actually deploy them soon enough. And through a month's exercise, we actually had 1,100 innovations came within a month, which were assessed by a very strong expert team over a month, and 31 were actually picked to then help with deployment. And I'm very happy to say that out of that, while this is an older number, almost 22 to 23 products are in market serving society. And that is actually a real outcome of what you say, investment in building innovations. This was possible, 1,100 innovations came around, and this were possible because last 10 years, we have invested in building innovation quotient. And that is something we have to do for helping the society in challenging times like this that we are facing right now. At the same time, CCAM is anchoring two of the largest COVID-19 diagnostics programs, again, working with startups and industry partners to build capacity in COVID-19. The reason I brought it out is that this all happened because when you actually have been helping bioentrepreneurship talent somewhere, and NBEC is a classic example, as I mentioned, to pick people from the ground on a platform which is completely unbiased and anybody can connect without any affiliation and go on the merit of the idea forward. This is NBEC. Previous years we saw in 2017, 2018, 2019, where we were able to actually physically meet and do it. These are the different times, but we were able to constantly, amplify the ideas across India, select them, and then nurture them across. And as Professor Vijay Raghavan said, that for a truly innovation-led entrepreneurship requires link between academia, entrepreneurship, and commercialization. And efforts like NBEC are those bridging gaps because we have on one side strong academic connections through various institutes, DBT, BIRAC, and other agencies. We have a strong entrepreneurship diaspora right here. And then the industry, which has become a very important, perhaps the guiding light for us to build those innovations forward. So this is very, very important, the way it is going forward. We launched the NBEC 2020 in September. We are now in December. And over the last 4 months, as my colleague, Dr. Niranjan Joshi said, that we have actually worked hard to take those 3,100 applications and step by step come toward today for a finale. And hopefully we'll have the announcement very soon. Again, we'd like to thank the industry partners who have actually grown exponentially as we have grown in the NBEC journey over the last 4 years. So as I mentioned, those innovations were from across India, including student teams. You can see that representation was there. This is very, very heartening because numbers are important, not to actually say that, okay, these are great numbers. Important is that we are able to attract more and more, curious minds, more and more innovators, and tell them that please share your ideas and build that further. Today, after all these efforts, we are here with the help of BIRAC to actually build this platform under BREC. and looking forward to work with you all. I would say, while the competition is built as a pull mechanism, it is not the end of the journey, as rightly said. I think it is actually a journey where some of the winners will be now hopefully handheld by the industry and then taken to the journey next one. I cannot end this presentation without thanking people behind the BREC and NBEC who actually made the entire journey possible, who have worked effortlessly in terms of for taking this forward and building it to the next level. Again, I would like to wish all participants all the very best. Thanks for the industry partners. Of course, many thanks to BREC, who has believed in the idea of BREC, believed in the idea of NBEC for the last 4 years. And hopefully we will continue to do better. And thank you all my colleagues here who are mentioned, Nandita, Niranjan, Bhavisha, Debolina, Rijesh, Ramya, and also Dr. Diwan and Dr. Shilpi from BREC for the continuous support to the program. Wishing all the very best to all participants and innovators. CCAMP is here to work with you. This is just a platform for us to come together, and I think journey starts now. We look forward to engaging with you. All the very best. Thank you.
Thank you, Dr. Taslim. Thank you for those introductory remarks and also your thoughts on investing in innovations, especially in the context of the COVID-19 pandemic. I think we all have gone through a very difficult year, and that is when we should start understanding and investing into the innovations. The time starts now. So thank you for those thoughts. I am now going to introduce Dr. Renu Swarup. Of course, for the Indian biotech ecosystem, she requires no introduction. Dr. Swarup is the Secretary of Department of Biotechnology, Government of India, and formerly Managing Director, BREC, where she was responsible for creating and operationalizing BREC. She was also a member of the task force on women in science constituted by the Scientific Advisory Committee to the Prime Minister, and also was awarded Biospectrum Person of the Year Award in 2012, as well as the National Entrepreneurship Awards 2017. Dr. Swarup will deliver her keynote address. Unfortunately, at the last moment, some urgent meeting came up with Union Minister Dr. Harshvardhan, and she couldn't be here in person, but she had sent a video message, and now I'm requesting my colleague to play it for this gathering.
Thank you, Niranjan. I'll just take a few minutes to play the video.
Sure.
Heartiest congratulations on this grand finale of the NBEC. I'm really pleased to see that there was such a tremendous response to this series of the NBEC, which was put out by the BREC Center or at CCAMP. The fact that you had a large number of innovators coming forward and put in their ideas for consideration itself is a recognition of the fact that we have a wonderful, vibrant ecosystem today. Congratulations, of course, to all those who have received the final awards. But I must say that each of you who's been an applicant to this NBEC is a true winner. The biotech ecosystem has been growing over these last few years, and BREC, which is the public sector of DBT, has worked to create this vibrant ecosystem along with all its partners. CCAMP, which is one of our very important partners, a big partner, and we have many other such big partners today across the country. Each of them have really put in a lot of effort to build this strong, vibrant ecosystem. We have more than 50 incubators today. We have 5 regional centers, each of them given the responsibility to work along with all the other stakeholders to see how we can attract the best minds, pick up the best ideas, and help them to go through this pipeline of taking an idea through translation to product. From finances to risk capital to technical handholding to mentorship to business mentorship, we try and see how we can offer all this to you. Because we are confident that you really are the ones who are creating this very strong enterprise for us. In these COVID times, we have seen how not only the biotech sector, but the startup ecosystem in the biotech sector has responded so beautifully. If in a short period of time, we've been able to bring in indigenous diagnostics, of course, our whole vaccine story, looking at therapeutics, it's because we have this very strong foundation. Similarly, our confidence of achieving the 150 billion bioeconomy in the next couple of years is based on the fact that we do recognize that there is so much of strength in these young startups. Today, I'm very pleased that you have a large number of industries, investors, national and global, who've come forward to engage with us in this very important mission that we have of how do we build a strong, vibrant biotech ecosystem. Congratulations to all of you. I wish you great success, and I'm sure Dr. Tasleem and his entire team and my entire team at BREC is going to be with you as you move forward. This whole ecosystem that we have created, in fact, each of the scheme that we have released, set up, is based on the feedback that we get from you. We would love to see you grow bigger, grow larger, and not just serve our own country, but be the global best. That's really what our ambition is, that India is going to be self-reliant and Atmanirbhar Bharat, not just for India, but for the world. And we are confident we can do it. We are confident that you will deliver. And that is really what our expectations are. This award today is a recognition of your effort, a recognition of your strength, and a very important step in that ladder of success that you are working towards. So enjoy this moment, feel proud of your achievement, and also work towards getting better and better. Our effort is not just scaling up the ecosystem, but sustaining it. And scalability and sustainability would only happen if we have all stakeholders working together and bringing in not just the large numbers, but the quality of innovation. And I think that's what we are trying to get. You bring in large numbers of innovators, they must be of quality, and we must be able to deliver at the global best standards. So good luck. Once again, congratulations, and we look forward to you to being much, much, much greater in your endeavors. Thank you so much. All the best.
Thank you, Dr. Swaroop. Atmanirbhar Bharat, not just for India, but for the world. Be the best in the world. I think those are the inspiring words that we take from her message here. And I'm sure that the NBEC participants, the finalists, the winners, and the whole ecosystem that has converged here today, we will walk under the guidance of Dr. Swaroop and follow these words. With that, I now have the honor of introducing another well-known face of the Indian biotech industry, Dr. Kiran Mazumdar-Shaw, the chairperson and managing director, Biocon Limited. She is hailed as the pioneer of biotech industry in India. She received two of India's highest civilian honors, the Padma Shri and the Padma Bhushan in 2005. Over the years, she has received many distinctions, not just in India, but across the world, including Time Magazine's 100 most influential people in the world, world's 25 most influential people in biopharma, and the only Indian on Forbes list of world's self-made women billionaires. Dr. Kiran Mazumdar-Shaw has been the constant presence in all the previous 3 grand finales of National Bioentrepreneurship Competition. She couldn't be here today, but she has sent her video message as the special address, and I'm requesting my colleague now to play the address.
Yes. लघु अब तो वो 15 मिनट में अनाउंस करने वाले हैं.
May I request to mute the other microphones, all
Hi,
the other
everyone.
participants.
I'm most disappointed that I can't join you for this year's National Bio-Entrepreneurship Competition, which promises to be a very vibrant and exciting event. And I'm delighted that this year's jury chair is none other than Nitin Deshmukh, who has been one of the finest and most astute investors in the biotech sector. I myself have been a recipient of his venture funding, and I have seen the way he evaluates, assesses, and invests in biotech companies, most of which have been outstanding successes. COVID-19 has created a huge number of opportunities for the biotech world, and I'm sure that all of you have come up with some very exciting and innovative ideas. This year has seen a surge in the number of bio-IT companies, bio-medtech companies, and of course biotech companies themselves in the vaccine area. So you can see that this is the most sought after sector. This is a sector that many venture funds are looking to investing in. And this year's NBEC program is going to be very, very exciting from a jury point of view. And of course, I wish all the winners all success in their efforts to take their ideas to the market. So all the best, and keep the biotech flag flying. Thank you.
Thank you, Dr. Kiran, for those encouraging words. We will, with your guidance and assistance and support, we will keep the biotech flag flying. Thank you. Moving on to our next speaker, Srimati Anju Bhalla. Srimati Bhalla is Joint Secretary DST and Managing Director BREC. She joined Central Secretariat Service, Government of India in 1990 and has varied experience from her work in the Ministries of Commerce, Industry, Culture, Women and Child Development, and Power. She is postgraduate from St. Stephen's College, Delhi University. BREC, as you know, has been a constant supporter of CCAMP, as well as one of the main partner member of the BREC Regional Entrepreneurship Center, under which this competition is being organized. I am now requesting Srimati Anju Bhalla to deliver her speech.
Thank you, Nilanjan. A competition or an event like NBEC actually is an event of winners. It's not just the participants or the actual awardees, but the people who have conceptualized this program, this competition, and the people who are carrying it forward on a day-to-day basis. I think you're all winners because it is your conviction, your teamwork, the partnerships that have been built over the years that have created, as ma'am said, a very strong foundation. And today I want to congratulate all of you, starting from Tasleem to Debulina, Nilanjan, all your team members, everybody at BREC, at CCAMP. And of course, there's the BREC team, which is supporting you constantly throughout the years and doing a wonderful job altogether. I'm a relative newcomer to BREC and to the biotech sector. My introduction has amply made it clear that I don't belong to this sector, but I've learned so much. And I'm tremendously grateful for this opportunity to get this window into the sector, to work with the BREC team. And I just consider myself very lucky, very privileged. So it's an honor to be here with you all today. And congratulations to all of you, to all the participants in the competition. Everybody who applied is a winner. And I'm sure that over the years, they will continue to win and add value to the ecosystem that is actually going leaps and bounds. It's growing so fast. That's amazing. I mean, within the span of a few months, so much has happened. It's tremendous. Sometimes it's difficult to absorb that it's actually happened in a period of a few months. So what the next few years are going to do, one can only wonder. So congratulations once again, and all the very best to all the winners and the awardees going forward. Thank you so much.
Thank you, Srimati Bhalla. I think your and BREC's constant support keeps us going, and we look forward to that support going forward. Thank you.
Thank you.
And now I have a pleasure of introducing one of our own, Dr. Manish Diwan. Dr. Diwan is Head Strategy, Partnerships and Entrepreneurship Development at BREC. He has about 23 years of global industry and academia R&D experience in pharma biotech sector. He's trained in pharmaceutics, molecular pharmacology, immunology, FIC and BIC drug discovery, preclinical, translational, biomarker research, and early clinical drug development. He has also been, the reason I said that he is one of our own is because as a part of the BREC Regional Entrepreneurship Center, as well as for the National Entrepreneurship Competition, he has been a close supporter, and I'm now requesting him to share his thoughts.
Thank you, Niranjan, for such kind introduction. I think the entire stakeholder network that we have for biotech entrepreneurship is so closely knit that all of us are our own and really part of the whole effort. I must congratulate CCAMP, first of all, for running the 4th edition of BREC. And the very fact that it is the 4th edition, it itself reflects that there's a constant pipeline of such innovative ideas which are there and which require 4 months of evaluation. So it itself reflects the merit and the quality that is getting built up. I wish that out of 275 startups and 40 student teams who have participated in the BREC 4th challenge, many succeed with their original ideas and convert and translate those ideas into tangible outcome, products, technologies. If the same ideas cannot be cannot convert as part of the startup journey, I think more evolved, iterated ideas would come out of these efforts. And all of you, with the collective strength of this ecosystem and the partners and the able support from this network, would get a lot of facilitation to move your journey and take it to a logical conclusion. As a part of enabler, as an enabler of the ecosystem, I think the BREC DBT's effort is to create provisions, for instance, for funding support. There are early stage programs like BIG, Sitare, eYuva, which facilitate at the ideation level. So I think many of you would like to engage into that. We also create incubation facilities like CCAMP is a BioNest incubator. Likewise, there is a network of 50 such bioincubation centers which are specialized to provide nurturing grounds for biotech, particularly biotech driven activities. There is a growing network of partners, and as we just heard, CCAMP is also a BIG partner. Likewise, the research institutions across the country which have the high-end instruments, the subject matter knowledge, like relevant industries as well as investors, and many of them have their presence also here. So that network, which is a knowledge pool, that is also integrated. We also make conscious effort to create opportunities and platforms. BREC is one such effort. BR, BRBC, BREC, BRBC, BRTC, 4 regional centers, they continuously make this effort all across the country to provide both national as well as global networking opportunities for all of us. I think the very fact that within the few months of COVID, we were able to come up with so many solutions. The entire network of this incubation facility, there are 125 solutions which were COVID specific, which got translated and entered into the market. So it itself suggests that there is a remarkable amount of talent as well as capacities which are sitting and which needs to be tapped and channelized. So through this effort of BREC, BRBC, the BREC, CCAM, and the entire stakeholder community which has been supporting this activity, that is a provision. So I must congratulate again to all who have applied, first of all, and the team at CCAMP and BREC, which has put together and make this effort a success. And I wish many among you would succeed. Few of them certainly would get acknowledged and receive the facilitation as winners. But I think as Dr. Sarup, Dr. Bhala, Ms. Bhala all suggested that this is a journey, this is an onward journey, and many would get succeed. Many would succeed in this endeavor. Thank you so much and wish you all the best. Thanks again. Thank you, Nilanjan. Over to you.
Thank you, Dr. Divine. Thank you very much for this, those kind words, and we will continue to work towards the more such editions for the years to come with your support and guidance. I know that everyone is probably very eagerly waiting for the announcement. So without much further ado, I am now handing over the proceedings to my colleague Nandita for the announcement of the winners, as well as some remarks from the chair of the grand jury. Nandita?
Yeah, thanks, Niranjan. Thank you. Yeah, I think everybody is waiting eagerly for the results, and we shall get to that very soon. Before that, would be delighted if Dr. Anand Deshpande, he was the chair of the student panel jury. As you know, this year we had a separate track for students and a separate track for startups. So we ran the student track in parallel because of the limitations of these online platforms. So if I can request Dr. Anand Deshpande, founder, chairperson, and managing director of Persistent, to make his jury remarks.
Thank you very much for inviting me. It was a real pleasure to be part of the jury process. I want to first congratulate all the participants of NBEC and especially the student participants that we saw. Their work that they had done was amazing. The videos were really good and their presentations were extremely good. So I want to thank them for that. I also want to thank the CCAMP team for all the work that they did, and my other fellow jury members, Sapna, Ravi Bhola, Naganand Doraswamy, who did a lot of work to help the process through. I must say that, you know, we had about 10 finalists who were there, and it took us a lot of effort to come down to the top 3 winners. We spent a lot of time, and actually most of the proposals were really, really good. So I must commend the students for the work that they had done. I just want to mention that, you know, all of you have good ideas and use this opportunity to get some feedback from the rest of the team. But don't give up even if you don't win the award. So I want to wish you all the very best. And thank you very much for inviting me. It was a real pleasure to be part of it. I'm equally anxious to know the winner. So I'm going to stop here and not waste too much time in getting this going. Thank you very much.
Thank you, Dr. Deshpande. The chair for the startup jury was Nitin Deshmukh, but really he has said that he really didn't want to come between the announcement and the his remarks. So without much ado, let me just check once more with Nitin if you would like to say something.
No, thanks, Nandita. I know everybody is anxious for the to know the awards. I really appreciate. Thanks for the opportunity to actually represent the very distinguished jury panel today. We had very great presentations, I must say. And most importantly, I'm very happy, of course, with the numbers that we've seen, 3,100 plus this year. And most important, the other thing was also the industry partners that we saw this year. Number of industry partners have increased, as well as I saw great enthusiasm from investors to actually pick some of the investments today, which unlike in the past, and we received great enthusiasm this time. In fact, I was also tempted to actually shortlist two in this year. Really goes to reflect the quality of presentations and the business plans that were presented in total this year. I was very impressed with all the videos that were sent, almost 18 business plans. I went through each one of them. I was absolutely top class, I must say. In fact, compliments to all the mentors at the boot camp to get them presented right, because that's very critical. It's also both from a content, number of slides, and the communication. I think all were very well done, I should say. Clearly, it's always a very difficult exercise for the jury to choose the best amongst the good, whatever. And of course, today was no different. But I think we need to sort of obviously look at which are the ones that stand out amongst most differentiated, I must say, amongst the good those are the ones that will eventually take the award. Clearly, I applaud all and celebrate these award winners, besides all those who made it to the finalists today. I really appreciate it. It was a great, I really enjoyed the entire presentation sessions. And I'm sure it will be a great encouragement to all and an inspiration to all the participants also at the same time. So congratulations to all the winners as well as the finalists. And great, thank you. Thanks for that. Thanks. Enjoy the day.
Yeah, thanks, Nitin. I was reminded of Jeffrey Archer's novel, First Among Equals. So I think that's what we are going to see. Let me just take some time to share my screen before I can. Is my screen visible?
Yes.
Yes, Nandita. Yeah.
Okay, I hope I'm able to get this right. So here we go. The Persistent Award. We are first announcing the student team awards. The Persistent Award for 5 lakhs goes to Tanish Sridhar.
Thank you, ma'am. Thank you, sir. Thank you so much.
Thanks, Tanish, and many congratulations.
Thank you, ma'am. Thank you.
This year, BREC also has instituted a prize. We chose to give it to a student team to encourage students. And the jury has decided that the BREC prize of 3 lakhs would go to Chandan Kumar Jha. You're on mute, Chandan. We would like to hear you.
Yeah, thank you. I'm speechless. So thank you so much for the award, and thank you so much for this entire experience. It was really a great experience for us and a great learning, and we'll continue to take our product forward. Thank you so much.
Thank you. Congratulations.
Thank you, madam.
Nandita, would you like to maybe in a line or so also let us know what the project and what's the product look like?
Yeah, sure. Maybe they can say it.
Yes.
Because I'm actually juggling 2 slides. So
Yes, I would like Tanish to take a minute and explain your product. Tanish, please unmute yourself.
Debulina, this is Chandan Kumar. Yeah, Chandan Kumar Jha. Dr. Diwan, he worked on intelligent virtual hand rehabilitation and assessment systems for stroke patients. Chandan is from IIT Gandhinagar, a student, a PhD scholar, and would be very much keen on taking his product forward. Chandan, if there are any other few points you want to say, you may add in here.
Hello, I'm sorry, my phone got disconnected. I missed a few minutes back conversation.
Chandan, is this Chandan?
Yes,
yes.
Chandan.
Chandan, Dr. Diwan from BREC just wanted to know a bit about your product. I have already mentioned what the product is. If you want to add in any few points, you may do so quickly.
Sure. Yeah, I'll just brief that in 2 minutes. So what we are trying to do is we are developing this gamified glove system for stroke patients, and we are also in the later phase. After 1 year, we are also going to build an exoskeleton specifically for stroke patients. So what happens is, in case of stroke, in most of the survivors, the fingers stop working because of the paralysis. So using our glove, we are providing a physiotherapy mechanism, and by which the patients will be playing the games, but at the same time will be also getting the feedback on their recovery. And this will motivate them to do these exercises again and again, and therefore recover much quickly.
Thank you. Thank you, Chandan.
Thank you, sir.
Nandita, over to you.
Yeah, so would Tanish like to just talk about in a line what he's doing?
Hello, ma'am. Hello?
Yeah, please quickly let us know what your product, what you're working on.
Yes, ma'am. Yes, ma'am. Sure. So ours is basically a water purification system. Hello?
Yeah, go ahead.
Yeah, sorry. Ours is basically a water purification system and a solid waste management system where we take in wastewater or polluted water and purify it with instantly to give secondary treated water, tertiary treated water, and drinkable water within 15 minutes. And the solid waste which we get, we make bricks out of it. From the revenue of the bricks or the profit from the bricks, we compensate with the purification cost, water purification cost, and so making our whole process a near zero cost wastewater treatment process.
Yeah,
Thanks,
Tanish. Thanks. Yeah.
Thank you, sir.
Yeah, then we have the third prize, which is contributed by K&S Partners, BioNeeds and Idea Spring. K&S Partners has contributed 1 lakh towards this prize. And the award for this goes to Abdul Jabbar Khan.
Abdul, you're on mute. Abdul, you're on mute.
Ma'am, am I audible now?
Yes.
Yes, ma'am. I am very glad to receive the award, and I would like to thank the jury and also the CCAM team who have encouraged my efforts and my team's efforts onto our project. We had developed a device for curtailing the residual ridge resorption, which is basically a gum massager, which is useful to the edentulous patients, the one who don't have a teeth. And by using the gum massager, they will be able to retain their bone quantity as well as have good denture intact. So this is what we had developed, and we look forward to incorporate our company as a limited liable partnership at this instant. And we are looking forward to produce such more units. Out of the grant that we received, we will be having some part invested into the machinery that is required for making the same. And this is what we are planning to do ahead. Thank you very much.
Thank you, Abdul Jabbar. Many congratulations.
Yes, thank you, ma'am.
We will now move to the startup prizes. These are not, I'm reading out the prizes not in any specific order as such. The first prize we have is from Origin Technologies, Discovery Technologies. And the prize goes to Cardiatech Medical Devices, Dr. Sugandhi Gopal.
Thank you very much. We are touched and we've been so happy to have Tasleem and the team's support in the last 3 years. I'm stunned and I'm very glad you chose us. Thank you very, very much. We'll work hard towards this. Thank you.
Thank you and many congratulations.
Thank you.
The next prize is by Biocon. And this prize goes to Cyclops, Mr. Niranjan.
Yeah, thank you so much for providing us this very unique opportunity to participate in this journey. And we are thrilled to win this award from Biocon. Thanks to Biocon, CCAMP, BREC, and DBT for this kind gesture. Thank you.
Thanks, Niranjan. Congratulations. The next prize is the L'Oreal Prize for Personal Care. And the winner of this is Avisa, Ravi Adgulwar.
Hello, am I real? Thanks, Nandita. Thanks, BREC, and thanks, L'Oreal, and all the people involved for choosing us. I would also like to thank my co-founder and the Bionest Kochi team for their continuous support for the last 3 years. Thank you very much.
Thank you and congratulations. The next award we have is the Ankur Seeds Award in Agritech, and this goes to Agritech Nikhil Das.
Hello, hope you can hear me.
Yes, Nikhil, we can hear you.
Yeah.
We can't see you though.
Oh, there's some issue showing like.
Okay, go ahead.
Yeah. Okay. Anyway, thanks for the award. Like, I appreciate the team. Like, it was a very well organized team. First of all, like, thanks to CCAM for doing such a big event. It's excellent, and thanks for the team. Like, and I congratulate my own team also. It was excellent. Thank you. Thanks a lot.
Yeah, thanks, Nikhil. Would you like to say what you are working on?
Yeah, we are working on a product which is on a seed classifier. Like, it's an image-based classifier system. So seed identification can do.
Okay, thank you and congratulations.
Thank you.
Yeah. The next prize that we have is the Pfizer prize, and this goes to Accord Innovations, Dilip Shankar.
Hi, ma'am. Thank you so much. Thanks for the award. And we are so grateful. And I should definitely thank CCAM team in all the ways who are supporting us.
Yeah, would you like to share what you work on?
Yes. So we have a device which monitors kidney function on real time to prevent hospital-acquired acute kidney injury.
Yeah, thank you and congratulations.
Thanks a lot.
We have the Anthem Biosciences Award next, and that goes to Adivo Diagnostics, Gitanjali Radhakrishnan.
Thank you so much. We are privileged to receive this, and thanks to BREC and everyone in the jury for organizing. So we develop a handheld device that can detect bacteria and fungus non-invasively on wounds, and we focus on expanding it to other segments as well. Thanks again.
Yeah, thanks, Gitanjali. And I would request you to stay on because the next award from IQVIA also goes to Adivo, Gitanjali.
Oh, that's that's amazing. Thank you so much. I mean, thanks again for all the support. I mean, yeah, same thing goes. Yeah.
Thank you and many congratulations.
Thank you.
We next have the Novozymes Award, and this goes to Prerna Tomke, Nanopearl.
Thank you so much. Thank you so much for the we are touch. Actually, we are working on the water purification solution, which is an all-in-one unit to remove the tedious heavy metals and microbes in the one unit only. So thank you so much, BREC and also the CCAM team for this award. Thank you so much.
Thanks, Prerna. And I think I will have to ask you also to stay back because you also get the Win Watson Award for your work. So
thank you so much. It's really, we are touched to receive these awards. Thank you. Thank you so much.
thank you and many congratulations.
Thank you.
We also have a Win Award for in maternal and child health, and that goes to Sascan Meditech, Subhash Narayan.
Hello. Yeah, thank you so much for the award. Yeah, it's a great privilege to be associated with CCAM and BREC. So we are developing this device to detect early stage cervical malignancies, and we hope that we will be able to make a change in the way cancer is treated in this country. Thank you.
Thank you and congratulations. We now move to the Investment Opportunity Award. Here, I must say that the jury discussions were really very interesting. And as I reveal the startups that have got these awards, I'm sure you will know why. So the Sangam investment opportunity goes to Agdi and Nanopearl. That is Nikhil Das and Prerna Tomke.
Wow.
Again, thank you so much for this award.
Thank you.
We are very much glad to receive this. Thank you so much.
Thank you.
Thanks a lot. Thanks a lot.
And congratulations. The next investment opportunity is by Enzia, and Enzia would like to meet further with Cyclops and Nanopearl. So Niranjan and Prerna.
Oh, fabulous. Thanks,
thank you so much.
Enzia.
Thank you so much once again.
Stunning. This is like a Sone Pe Suhaga moment. Thank you.
This is really an amazing feeling. Thank you so much.
Thank you and many congratulations. Indian Angel Network also has picked 3 opportunities for investment. Quizzens, Srishti Batra, Cyclops, Niranjan, and Cardiatech, Dr. Sugandhi Gopal.
Thank you so much. This is a great opportunity and looking forward to talks.
Thanks, Ian. Looking forward to have you along.
Yeah.
So thank you.
And Sugandhi Gopal, Dr. Sugandhi Gopal as well for the Indian.
Yeah. Thank you. Thank you so much.
The Molecular Connections investment opportunity goes to, again, Cyclops and Cardiatech.
Thank you.
Whoa, phenomenal. Thank you.
Many, many congratulations.
Thank you.
The Social Alpha investment opportunity goes to Godam, Kalyani Shinde.
Hello, thank you so much, Social Alpha, obviously to the entire team, but it has been a long time I wanted to work with Social Alpha on something. We've also had discussions with Manoj sir and on, but I think this is great. Thank you so much. Thank you, everyone.
Thank you. The other, another investment opportunity comes from 91 Springboard and their syndicate committee. And they would like to further meet with Quizzens, Agdi and Godam. That is Srishti, Nikhil Das and Kalyani Shinde.
Thank you so much again for showing this great interest. Really looking forward to talks.
Yeah, thank you. With this, we finished the investment opportunities, but then, as I said, the jury presented us with a surprise, and happy to announce here that Kotak Private Equity Nitin, who represents Kotak Private Equity and is in fact a founding member of the Kotak Private Equity practice, would also like to propose investment opportunity, and that goes to Cyclops and Adivo. So Niranjan and Gitanjali.
Thank you so much, Nitin. It's a privilege to have this commitment from you, and we eagerly look forward to your guidance. Thank you so much.
Yeah, Nitin. Thank you so much. I would be happy to interact with you again. And we have some interesting updates, which I'll be happy to share with you. So thanks again.
Thank you. We also have a digital health bootcamp award. So the winner of this gets to participate in a bootcamp conducted by Health Venture. And for this, the jury has chosen, it's not a startup, Sanjeev Sambandhan. So Sanjeev, many congratulations.
Thank you very much. And firstly, thanks to all of you to give me this opportunity. It's been a very interesting journey, and many thanks for this recognition. It's very encouraging. Look forward to it.
Thank you. And as I said in the initial, when I started, that we have this year not one but two surprises. The first surprise was the Kotak award, and the second surprise is from CCAM. We would also like to join the party. If I can request Tasleem, this award also goes to Sanjeev Sambandhan. And Tasleem, maybe you would like to say something about why the jury chose Sanjeev for the CCAMP award.
Yeah, no, I think Sanjeev's work that he should talk about in a minute is a phenomenally exciting and futuristic. And CCAMP doesn't want to be anywhere away from there. We want to be right there. And this is the award for us to start the journey together, Sanjeev. You know it, that we have done it for the other one as well. So we want to be with you. So just please, if you can share your thoughts, early ideas that we want to build further with you.
So yeah, thanks a lot, Tasleem. I'm sort of speechless, but yeah, this idea which I presented today, I see a lot of potential. I have also worked with all of you before on my, the other startup, which is Open Water, which has taken off very well. But this idea, I see a lot of potential. It's in very early stages, but looking forward, I'm looking to work with pharmaceuticals and probably get this out as soon as possible. So it's a very high power drive that I want to put behind this, because looking at the pandemic we had, this sort of fits the picture very well, wherein you need this, non-interactive diagnostics and so on. So I think this is this idea of this ingestible or edible pill is coming in at the right time, and I look forward to working with all of you on this.
Yeah, if I may say so, what Sanjeev is attempting to build is an edible pill for diagnostics, which can actually do a GI tract diagnostics and so on. So it's a phenomenal idea that he's building on. So, that's fantastic. Thank you.
Thanks a lot.
Thank you. I think with this, I won't say that we've come to almost to the end of the program, but it's always a new beginning, and we are looking forward to the next year as well. But I would like to ask my colleague Bhavisha to make the closing remarks.
Oh, hello everyone. On behalf of CCAMP team, I would like to thank you all for being part of NBEC 2020 Grand Finale today. I extend a really hearty vote of thanks to guest of honor, Dr. Renu Swarup, Dr. Kiran Majumdar Shaw. Although they couldn't come, but they were kind enough to send the message. And I also thank Srimati Anju Bhalla and Dr. Manish Diwan to be here and grace this occasion. I am immensely thankful to all the NBEC partners for their time and contribution. All partners have been very supportive and proactive to make the NBEC a success. We anticipate their support next year as well. I am inordinately thankful to all the jury members, reviewers, mentors for sharing their time and expertise for NBEC and helping us to choose the finalist cohort. Participants have gained a lot from your comments and feedback. We have received a very good feedback about all the comments from all the jury members and support from the mentors. Also thankful to all CCAMP friends for helping us in social media outreach of NBEC in nook and cranny of the country. It was, as you all know, it was very difficult this year that like with COVID, we couldn't go anywhere for outreach, but like I have, me and my colleagues, we have written to almost all the friends, families and all CCAMP friends. We have bothered you by WhatsApp messages, social media, that kindly promote NBEC. So thank you all for doing that. And lastly, I would like to say that an event like this cannot happen overnight. It's a lot of work from whole of the team. At CCAMP, we are very fortunate to have a wonderful dedicated team to successfully run this flagship event. So thank you, Dr. Taslim, Wing Commander James, and CCAMP team for being supportive and proactive. And thank you one and all for joining today. Have a good day. So we will close this meeting in about 5 minutes. We will be happy to have your feedback in the chat. Thank you all.